discussion what we had yesterday. The two variants of real-time PCR. One is the fluorescent dye-based qPCR chemistry, then another one is the Tachman probe-based qPCR chemistry. I think you understood that. I think I oversimplified it so that you all understand that the secret behind how the qPCR really works. Any doubts on that before I move on? So the fundamentally one is non-specific in nature and another one is specific in nature. Just a quick recap of what we did just to rewind some of the things. So this is a dye-based quantitative real-time PCR. We use an important dye, very popular now, that is cyber green, right? So what is the character of cyber green? Anybody? It binds in the minor groove of a DNA double helix, right? As the amplification progresses, you get a progressive accumulation of amplicons. Amplicons are nothing but double-stranded DNA. They would carry the minor grooves and your cyber green is going to stick inside. And as the DNA accumulates, you are going to see a progressive increase in the fluorescence. That's what you are going to see in the machine. Yeah, different variants we talked yesterday. So cyber green 1, 2, and a lot of variants are there. Yeah, this is cyber green chemistry. So remember, the dye is unbound at 0 hour. As the cycling is progressing, the unbound dye is going to be bound in the minor groove of DNA. Okay, so that emits the fluorescence, and that fluorescence is captured by the camera, the spectrophlorometer, that is placed just above the machine. It is something like it is monitoring the reaction, what's going on in the tube, and it captures, and at some point of time, fluorescence is so much, and you start seeing the typical curve raising up, right? So that when it meets the so-called basal level fluorescence, that point is what is called as CQ value or CT values. We'll see a little bit of that today. That is all about the so-called cyber green based or fluorescent dye based qPCR. Okay. Advantages are quite easy, not too much complications. You need only the extra dye and a machine. Otherwise, it is a typical normal PCR. The disadvantage, of course, the non-specific nature of this dye. It can bind to any minor groove, no matter what the DNA sequence is all about. So that conveys a very important point. You may go wrong in your interpretation if the primers are not very specific. This is probe-based. It is slightly different from the dye-based. At 0 hour, you are supplying a probe that is having a reporter, we call it as a fluorophore, and a quencher that is going to silence the fluorescence of that reporter. So separated by nucleotide sequences. Who is supplying the nucleotide sequences? It's you. You are designing the probe. Probe means the nucleotide sequence. And that it's as equal as the primer designing. There is no difference. Okay? You are supplying, the company will manufacture that probe for you. We call it as a probe because using that particular sequence attached to the two chemicals, you are probing what's going on there. For that reason, we are calling it as a probe. So there is a reporter, there is a quencher. The property, the unique property of this particular probe, it doesn't fluoresce when it is having a quencher in the close proximity of the reporter. If that is there, then it is not going to give any fluorescence. So if you want to see the fluorescence, the quencher has to be removed. So that is what the whole story. I already talked about 5' to 3' exonuclease activity of the tic-tac polymerase that is going to remove the, or rather separate the setup into, reporter separate and quencher separate, and the bound reporter is released and it is getting accumulated in the reaction mixture. It's quite different from the so-called fluorescent dye-based qPCR, right? There it is a bound dye you are monitoring. Here it is an unbound or released dye you are monitoring. Yeah, this is how it is. The cartoon I already put it yesterday. This is a cartoon, fluorophore and quencher. And what happens is it goes and binds to the complementary sequences in the template, and it is carrying the reporter and quencher. In the first step, it is getting segregated into two, and the TAC enzyme is going to come into play. And as it adds the nucleotide on the other end, in the front end, it is going to release the reporters and quenchers. And finally, you are going to see something like this, and the reporter is getting accumulated in the reaction mixture, and finally, it is going to give you a progressive fluorescence because the dye is getting accumulated in the reaction mixture. Any doubts on this? I just repeated, and some of you may be knowing or may not be knowing, but just to clarify. We'll take it forward. This is highly specific real-time PCR, adaptable, very much useful for pathogen detections where the false positives are not tolerated, and you can use it comfortably in that. Okay, from here on we will move ahead of what you did yesterday in a theoretical account. I think what you practiced yesterday is cDNA-based qPCR, correct? You use cDNA as a template, not RNA, right? That is a two-step qPCR, isn't it? Can you tell what you did yesterday? Anybody? What you practiced yesterday? What was supplied to you in the form of template? RNA or cDNA? cDNA, okay. This is a cartoon. It's quite simple. You can isolate RNA from the sample, and that RNA can be preserved for a long time. You can convert into cDNA as and when it is needed, and you can go for PCR. Okay. And there are some applications you don't have to preserve your RNA. You can straight away get into the quantitative real-time PCR, where you can go for a single step real-time PCR. And single step is quite easy. You can add one more enzyme, right? That is a reverse transcriptase in the same reaction mixture, and all in one. It's going to do the amplification. Okay. There is not much big difference in terms of actual experimentation. The fundamental difference is something like this. In one step, you have everything in the same tube. In addition to the conventional PCR recipe, you are going to add reverse transcriptase enzyme. Instead of a DNA template, you are going to add RNA, and also you are going to add the fluorescent dye if it is dye-based qPCR. Okay, and set up the normal PCR, and I set up the PCR in the real-time machine, and you start scoring the results. This is two-step. What do? You have the RNA, you prepare cDNA, and you can preserve those cDNA in your lab for long-term use. And as and when it is needed, you take it out and do the PCR. Here the difference is you don't add reverse transcriptase in the PCR mixture. Rather, you are directly adding the cDNA as a template and add your fluorescent dye and go for the real-time PCR. Okay. Requirements, what all you need if you want to really do a decent real-time PCR. Of course, you need a machine. So the machine what we have in the Discovery Lab is from Biorad, CFX96. I think those who have not used it and go and see and start practicing it. And you need a template, of course. What is template here? Template is your sample. It can be anything. It can be from the microbiome perspective, maybe a sample which is exposed to microbiomes. and collected in a different timelines. The sample what was given to you yesterday was something like that. A rice plant which is exposed to certain bacterial communities, and the samples were collected in a different time points, I think 0 and every 24 hours up to 5 days, and you are monitoring the activity of a defense contributing gene, and you are trying to make some interpretation whether microbiomes can activate some of the defense pathways in the plant. So template is very important. The follow-up lecture is about that only, the biological sample preparation. Primers, of course, very, very important. So we have a primer designing class also this afternoon lined up. We will teach you how to design a primer for PCR as well as qPCR. Okay. And you have a probe. If it is a TACMAN or probe-based, you have to design one. And some extra work, you have to design again, find out the specific sequences from the gene, and you order to a company to synthesize the probe. They will be adding the fluorophore and quencher, and they will supply to you, and much like what you are receiving from the company in the form of primers, same. And the DNTPs, of course, all of you must be knowing, the cofactors, enzymes, and the buffers. So except the template, all other things are purchased, correct? So only template is the one you are going to prepare in your lab. Depends upon the biological questions what you are having. Cycling conditions, I really don't want to narrate it because all of you must be knowing the 3 broad cycling conditions. There is a denaturation, there is a annealing, there is an extension, and you repeat it for certain duration of cycles. When you stop it, there is no difference. It is much like a conventional PCR, whatever you are doing. This is what I have been telling. There is always a ground level expression, or what is called as a basal level expression. The machine cannot read it. The point at which you start seeing the machine is able to read that, and that point is what is called as a threshold value. It's popularly called as CQ or CT values. And the data analysis is all about conversion of these CQ values into interpretable parameters. For example, fold change. I think the team will teach you how to do that. Okay, the CQ value is transformed into another easy to understand parameter. That is what is called as fold change most often. Okay, and there is a statistics in play. So we will be teaching how to use certain statistical tools to make a significance of the fold change. Okay. Yeah, this is a threshold line. There is a few definitions. You can read that. Baseline of amplification. Baseline of amplification plot. You can just read it. Initial cycle of PCR. There is a little change in the fluorescent signal. An increase in the fluorescence above the baseline that indicates detection of accumulated PCR product. Okay, so this is very important point. And threshold line is a point at which the reaction reaches a fluorescent intensity above the background. It is set in the exponential phase of the amplification of the most of the accurate for the most accurate readings. So the CQ values is something indicates that there is a amplification is happening and the reaction is working. If the threshold load is not reaching, most likely the PCR is failing. Cyclic threshold, and the cycle at which the sample reaches a threshold level, and most often the cyclic threshold value 40 and above is considered as a negative result, right? So, or rather no amplification is happening. So these are some of the thumb rules in qPCR. Another terminology you encounter is a melting cows. I'm trying to link this with the deficiency of the so-called fluorescent-based qPCR, where I already told the specificity is always people doubt because you never know whether the fluorescent intensity is increasing because of the target amplification or the non-target amplification. So in order to rule out, you have to do this extra work. It's not extra work, it is the same, the follow-up of the same qPCR. This is what is called as MELD curve analysis, right? So what I showed here on the screen is a typical single target amplification without any background amplification. Can anyone tell if there is a background amplification because of poor specificity of the primer, how this graph is going to look like? Anybody? You can imagine and tell this. If there is only one target and that is amplifying without any background amplification, you get something like this, a single peak, right? If there is another target which is also amplifying the background, you start seeing another peak. So you normally see a multiple peaks. Remember, if you see multiple peaks in MELD curve analysis, in all probability, your primer is not very specific, okay? To gain confidence in your data, you have to provide this MELD curve image whenever you are going for a research publication from your work. Without MELD curve data, we cannot say that your qPCR targeting a specific gene is very specific for that particular question, biological question or whatever experiments you are doing. Okay? Try to do that. What is MELD curve? Change in the fluorescence when the double stranded with incorporated dye molecule melts into single stranded as the temperature is raised. Usually it is done at a temperature of around 80 to 85 degrees Celsius. Okay? Now most of the qPCR machines, this MELD curve analysis is built in and you can do that very instantly. Okay, how it happens when the double stranded bound with the cyber green, one dye is heated, a sudden decrease in the fluorescence is detected at the melting temperature. Okay. The analysis part, I think you have to do with the data sets. I think Shanu, Javed, I think you have the data with you and you just demonstrate to the students how the data should be analyzed. And there are two terminologies or applications of qPCR. You might have heard about it. One is absolute quantitation and another one is relative quantitation. Okay. We'll have a little discussion on that. The level of expressed gene may be measured by absolute or relative quantitative real-time PCR. Absolute quantitation relates the PCR signal to the input copy number using a calibration curve, while the relative quantification measures the relative change in the so-called RNA levels, depends upon the experimental circumstances. Okay, not a big deal here, very simple, just follow the slides. The choice of the tool is depends upon the biological questions what you have in your mind. Okay, simple. Absolute quantitation is something normally we use in pathogen load estimation, viral load estimation, copy number estimation, right? Here you need to have a standard curve. It's nothing but any other chemical analysis you are comfortable with. For example, spectrophotometer readings, you have a standard curve, right? In known concentration of maybe the compound what you are detecting, you generate the values and plot in a curve with a very regression coefficient very closer to 1, so that you have a very high level of confidence about the calibration curve. Exactly same here, right? You supply a measured quantity of the so-called template and do PCR, generate the data points, plot it in the curve, make a calibration. It's usually a linear with a regression coefficient of 0.99 something, you'll get very easily, not a problem. Do that, then you supply your actual test samples. Then from the calibration curve, you can decode how much of copies of your target is there. It's very easy. Relative quantification is the one what you did yesterday. Okay. Normally, you take a gene which is not affected at any conditions of your experiments. For example, there are genes in plant, it is not affected by the microbiome treatments, right? So that means whether you give microbiome or don't give microbiome, their expression level is always same. So the tricky part here is you have to find such a gene, and usually the housekeeping genes are the one which qualified for this kind of analysis. Okay. What was the gene you used yesterday? It's actin. Actin. Okay, actin or actin. So there are plenty of choices are there, but those who are doing this kind of analysis, you have to standardize the amplification of those the so-called reference gene beforehand, okay, and gain confidence on the reference gene. There should not be any significant changes in their expression under the experimental conditions. Ensure that, okay. Yeah, absolute quantitation, it requires the construction of an absolute standard curve for each of the target gene. It is directly applicable in pathogen load estimation, copy number estimations. If somebody is checking or testing a sample for a presence or absence of a pathogen, or testing how much pathogen is there in a sample, and you must be having the primer which is very specific for that pathogen, right? And use that primer. You use that primer and also the DNA, a known quantity of DNA, you make a standard curve. Okay. So that is the first one. The standard curve is based on the serial dilution of the sample with a known copy number. Okay. And again, the CQ value, whatever you are getting for each of the known concentration of your template is plotted, and that gives you the typical what is called as standard curve. And from the standard curve, you can decode how much of pathogen is there in your test sample. It's very easy to do that. Relative quantitation is something where you are comparing your target gene to another gene in the same plant which is not affected by the experimental conditions. It's very simple, right? So actin, we know that it is not affected by the microbiome treatment. Right? No matter whether you give any quantity of microbiome load, actin is not going to be affected. So you use them as a reference gene. With reference to that particular gene, how much is your target gene amplification is happening, all right? So what was the gene you used yesterday? It is a chitin elicitor receptor kinase. And we know that chitin elicitor receptor kinase is something which is under the influence of microbiome. For example, if you give more of a microbiome, this CERK gene expression will be elevated, right? And if you don't give, maybe a normal expression, or sometimes some microorganisms can reduce the expression of this particular gene, right? So we do not know what kind of results you are likely to get, but you have to do it. So with reference to a gene which is not affected by the treatment, you are comparing your test samples, okay? So remember, whenever you do this relative quantitation, every time you have to use the reference gene parallel to your test sample. And this is a very important point. Many times you feel like, okay, I have done on my first data sets. Rest of my data sets I will not use because I can use that particular data. It won't work that way. Every time you have to use the reference gene parallel to your test sample. Okay, so that, of course, that would increase your experimental cost, but there is no other option. And that is true even with absolute quantitation. It's not like a standard curve is made once and the rest of the period you can use your test sample and you can decode the values from the standard curve made in the first attempt. No, it's not like that. You have to do this standard curve every time whenever you are doing this, what do you call, estimation of a pathogen by absolute quantitation. Remember these 2 points. You have to, every time you have to use the reference gene, every time you have to use the standard curve. Yeah, there are few points I think the team will tell you. Some of the housekeeping genes, which is very popular, there is 18S ribosomal rRNA coding gene, GAPDH, actin, and the list really goes. There are several options are there. But whatever options you are using, don't go with the name. Better you generate the data and see it is not affected by the treatments. Then you settle down. The best bet here is you try with a couple of genes or 5-6 genes, and you settle down for one which is really giving you a stable results across your experiments. Okay. Yeah, this is absolute quantitation, right? Yeah, and just rush through these slides. It's quite easy. This is a typical standard curve. You just can see here. And what is plotted here is the CQ values versus the template quantity, right? If you give more of a template, and your CQ value will come very quickly. I'll just skip it. These are all some of the calculations. The team will handle that. Yeah, some consideration for absolute quantitation. It's not very complicated, very simple setup, easy mathematics because you have the standard curve already prepared. You require a standard to be run every time you are doing qPCR, and the starting concentration must be measured. Relative quantitation, this is very important. Whenever you do relative quantitation, you please put this 4-checker table, and you identify what is what in that table. For example, 4 terminologies are here. One is a calibrator, test, and a target, and a reference. Okay, I'll just mark it up. It's very easy to understand. Calibrator is a mock plant. That means it is not exposed to the treatment. Okay, the control plant. Okay, there is a term for that. It's a calibrator. And you have a test plant. That is the one you inoculated with some microbiomes or bacteria of fungi, whatever, right? And target is the target sequence that you are monitoring. Okay, it could be CERK yesterday, or it could be any other gene. I think PAD4 also you have taken yesterday, PAD4, and it could be anything of your choice. And reference is something which I already told you, a housekeeping gene which is not affected by the treatment conditions. Okay, so I just plotted it here. A mock plant, housekeeping gene, that is a reference. Test is an inoculated plant, and the target is an effector gene in the case of pathogen, or it could be if you are handling a pathogen here for quantitative PCR. If it is a plant, it could be a receptor, or it could be a gene which is upregulated under the influence of microbiome. So remember this checker plate and plot it. That will give you some better ideas. Yeah, these are all some of the normalization steps. I think the analysis they will tell you. I'm just skipping this slide because these are some of the outdated calculations people used to do. Nowadays, everything is computerized, and the qPCR programs, the different companies are supplying directly to you. So it is a kind of, the machine itself will give you whatever data you want finally, right? But nevertheless, at least try to understand these are all some of the statistical, I would say, the methods for analyzing the qPCR. LIVOC or double delta CT, I think most of you might have heard about it. Delta CT method using a reference gene, PAF-L method, that is another method. Yeah, I'm just skipping it because there is no point in get into the finer details of that. Now things are simplified. Everything is now, the softwares for analyzing the data is already integrated in the program, and you can straight away take the readings. And for relative quantitation, of course, you don't need to know the starting concentration of your target. You are going to just see in relation to a housekeeping gene how your target gene is expressing. It is a kind of upregulation or downregulation, or what do you call overexpression, or what do you call repression. All those data will come. Okay. Of course, this is very, very useful for microbiome research. I think our group is routinely using this as a standard tool in the lab, just to prove that the microbiome applications can induce the plant gene expression or alter the plant gene expression more towards contributing defense, right? What is called as immunocompetence by the microbiome treatments. Yesterday also you heard about that particular paper appeared in Environmental Microbiome. We have used several genes in that particular paper. Very useful for plant or pathogen gene expression due to the presence absence of each other. It's very, very simple way of putting things. Yeah, of course, these are all some of the steps. Okay, just skip this. There are a variety of applications. We have already talked about it. And gene expression analysis, detection of mutant genes, copy number measurement in a transformed cell, pathogen detection or viral load estimation, and SNP analysis. And it really goes, and a lot of other applications are also there. I'm just not covering it here. These are some of the machines which is popular. And what we have in the Discovery Lab is from Biorad, CFX96. What we have in our lab is Light Cycler 96, and there are a variety of tools are available. So that's all the spillover lecture from yesterday. And I'm sure since yesterday we have been talking about it, you also practiced it yesterday, and today in the forenoon we'll be seeing the data analysis. With that, we will conclude the qPCR, one of the very, very important tool in microbiome research. You can really use it very comfortably in future. Okay. Yeah, I'll stop it here. And are you ready now with the? Yeah. Okay. You can stop it for a minute. Please, questions. Better go with the literature. For example, yeah, there must be a starting point, right? And shortlist them, and depends upon your time. Yeah, I didn't talk about that. Of course, the temperature regime or temperature conditions for amplification should be same as you can't have two different temperatures, right? It doesn't work that way. No? It should be a common, it should respond to the common recipe and also a common temperature conditions for amplification. That is also, it needs some amount of standardization. Yeah. Any more questions? Maybe in analysis you may have some more questions. Yeah. You can put on the light, no problems. Yeah, please. Yeah, should be same. Should be same, not it will be same. It must be same. It should not be affected. If it is affected, then your interpretations may go wrong, you know.
So, in my talking, you said that sometimes mouse changes with the application of microbiomes.
A skipping gene?
Expression changes with the application.
Yeah, you should not use it again. You have to settle down for a gene whose expression is not at all affected by the treatment. Forget about microbiome, even the treatment, any kind of treatment. Yeah. Yes, please.
So, for example, if I'm selecting from any sort and target gene, and I
Excuse me?
If I'm selecting some 8 to 10
Okay.
target genes, and the temperature in labeling of one particular housekeeping gene is matching with some 4 or 5 target genes, and the other 4 or 5 target genes, if
yeah, I got it. You need to have a common annealing, that's what I told. It is preferable to have a common conditions, whether it is a reaction conditions or it is a recipe conditions. If it is not there, it will add up to your work, because for every different gene, you have to use a different reference gene, then it's going to be, that is also doable, because after all you have to do separately only. It is still possible, yeah. Yeah, it is. Yes, of course. Absolutely. When you write a manuscript, you indicate all those things. That's very important. If it is a common reference gene is there, it is always better, but most often it need not be, right? So maybe you are having around, I think we are also facing the same problem because some 4 of the reference gene, the annealing temperature is different. The rest of the 5, it is a different temperature. Those things are bound to happen. So that is what the initial optimization of your work is very, very important. Not only for the reference gene identification, but also the reaction conditions for several targets. Yesterday, one of the group, the transcriptome validation they did with 50 genes. Just imagine, I do not know which group, I listened to the lecture. So 50 genes they have standardized. In the sense, of course they have standardized. And with a 50 gene expression pattern, they validated the transcriptome data. Right? Normally we don't do that here. We do around 10 genes normally. And 50 genes are all really very good if somebody is doing that. But just imagine for all those 50 genes, how much of optimization they would have done it. It's a great, excellent work yesterday. Anybody? Okay. We will move on to, you have the slides already? Yeah, okay. We're going to put it here. Okay, fine. Now the team will tell you how to do the data analysis. CQ2.
Doing good. So thank you so much, sir. Sir has explained every detail which we need for qPCR analysis and how to apply it, the application of this. So I would just give a brief of something like that also. I'll be telling you what are the formulas which we need for the analysis part, how to convert the CQ value or the CT value, or some machine also gives CP value to convert into the fold change. And then Javed sir will be telling you practically how to do it in the Excel. So I will start. So what we did yesterday, or you can just write it also for your own benefit. We first isolate RNA from the sample which we have, the biological sample which we have. It can be a plant shoot, anything. And then what is this? Then we convert this RNA into a cDNA. And what we generally see in the protocols which we have from the kits, which from Omega or Kaizen, we take 3 microgram of RNA sample to convert into a proper functional cDNA. Then from cDNA quantification, using a fluorescence dye in a qPCR master mix, we have that fluorescent dye, and through thermocycler we can get the quantification of those cDNA, or I can say RNA expression. Then the next part is analysis. So how can we use this qPCR? I will just go broadly. According to the papers, when we do some transcriptome analysis, they need to have a qPCR validation. So what we generally do in our lab is that first we get how many genes are, getting expressed in our sample. So what we take the top 10 which are positively regulated and the down 10 which are negatively regulated or down regulated. So we select those genes, then we design and the primers and get them synthesized. Then we take the same source, same sample from the RNA which we have sent to the company for outsourcing. That same RNA sample we will take, and then we will use these particular genes to validate our transcriptome result in the form of log2/4 change. Second, what we did yesterday, we take our samples, get RNA, convert it into cDNA, then use our defense gene, or as sir explained, you can even look for the presence or absence of some cultures, and then we can get the results. Okay? Now, this is how actual analysis, after your slide cycler part is completed, you will get a screen which will show you all these details, that what was the conditions of the cycle which you have ran. So this will tell you what was the temperature at what step. So I will go into a deep, I guess it is quite visible. Is it right? It's visible? If not, just tell me, okay? So generally in qPCR we have at least 4 steps. First is pre-incubation, next is amplification, third is melting, and the fourth is the cooling. For pre-incubation, we have the temperature for 95 degrees centigrade at 300 seconds. This is generally a normalized thing. Even if you want to make changes, you can make changes according to the literature you are taking reference from. If not, it's better to go with the standardized one. Next is the 3-step amplification, which we generally know denaturation, alanine extension. In that, I guess it's visible, 95 degrees centigrade for 30 seconds. Now, the thing which you need to change, which you need to know, is the annealing temperature of your genes. So you can make changes. For what we did yesterday, it was 58. So 58 degrees centigrade for 30 seconds. You can make changes into that before your thermocycler process. Okay, next is 72 degrees centigrade for 30 seconds. The next step is the melting. That is a very crucial part because, this is where the probes and everything is there, or your dye is there, it will start giving fluorescence. Okay, this part is also divided into 3 temperature sets. That is 95 degrees centigrade for 10 seconds, 63 degrees centigrade for 60 seconds, and 97 again for 1 second. Last part is the cooling temperature. What we generally see in our normal PCR, what is the cooling temperature? Can anyone tell me? 4. In this case, it is 37 degrees centigrade. So the conditions you have to normalize. I would again say normalize. This is the standardized one, so I am telling you that. Next, what will you get after you have thermocycler part is over? You can get different type of graphs. The one which we consider are very much important and give us some idea of what is happening is melting curve. I guess sir has explained what is melting curve. I won't go into that. And the third is you will also get a fluorescence curve. Fluorescence curve will actually tell you whether your thing is getting fluorescence or not. It is getting, some data you are getting from that particular well or not. Every line which you are seeing here represents every well which is considered for your analysis part. Okay. Then, as sir has explained, there are two types of analysis: relative quantification as well as absolute quantification. What we did relative quantification. Okay. For that, I guess the next one is that your heat map showing the CQ values. Okay. Towards this side, you have CQ bar plot also. So you can actually visualize, okay, my things are getting fine or not. Okay. The next one, the down one is the part which you need for analysis. It is a tabular format and you are going to convert this format into Excel. Okay, every machine has its own software. This is from Roche, Light Cycler 96, okay? But the thing you need is CQ values. Every instrument will give you that. So you just need to take it, convert it into Excel format for your own quantification, for your own analysis, and then you're done with it. Next is how to do the calculation. What is the status behind that? Okay, this is a sample which I have depicted in a very simple language. Okay, so you can see that in row 2, 3, and 4, it is unknown. Okay, and 5, 6, and 7, it is GAPDH. That is your housekeeping. Okay, so here what they have taken, there are two types of sets. First is the calibrator. Second is the unknown. That means treated and non-treated, I would say. Okay. Next, we have GAPDH as his housekeeping gene, and the other one, some other gene, unknown gene in your case. You can say that. Okay. First, we need to calculate the delta CT value. How you will do that? You have your CQ values. In general way, what we can do is we can do what we did, like we did replicates, triplicates, what I can see. Okay, for every gene we used 3 wells, right? So you will be getting 3 CQ values. First you calculate the mean of those values. Okay, after that part is done, now it's for further statistical analysis. You can calculate center deviation also, standard error also you can calculate for your, good biostatistical data. Then you go for delta CT calculation. What you need to do is you need to take your CT value, target value minus your CT reference. So in this case, what they have done, they have taken GAPDH CT value also as a reference one and this HCM 2J2 value as the target one. So this is how you will get your delta CT value. Am I clear till now? Okay. Next is to calculate delta CT value. For that, what they have done, they have, delta CT value they have calculated for this calibrator also. You need to do for all the sets which you have, okay. Then is delta CT value. What is the next thing? They have taken delta CT value of your test sample and delta CT value of your calibrated sample. I guess it is quite clear. If it is not, then I will explain more. Actually, it is already there. You will be having CQ values. Okay, from CQ value, delta CT value you have to calculate. In normal language, I will explain. Delta CT value, you need these two values. For this delta CT value of calibrator, you need these two values. Okay, they are already there. These are the main CQ values. Okay, solved? Okay. Now, as you have your delta CT value, Next, we go for log2 fold change. For that, as you already have your delta delta CT value, the formula which we apply in Excel is equals to power bracket of 2 fold change, we write 2, comma, your delta delta CT value. Then you will get your log2 fold change. Is everything clear? It's better you write this formula because sometimes people get confused how to calculate. Should I repeat the formula? Okay. Equals to power bracket open 2, because here log2 fold change is calculated 2, comma, your delta delta CT value, bracket closed. Then what you will get? You will get the log2 fold change of this HCM2J2 in reference to your GAPTH as well as your untreated and treated. Okay, how much fold change was there in comparison? This is the theory behind it. So what we generally do to represent your data in a very colorful way, we generally use graph pad prism for that. Okay, simpler graphs are there, very informative. You can design your own things, use colors, you can make good graphs using graph pad prism. I will explain one. This is what I got. You can use these type of graphs for your papers. It will be very nice, what I have seen till now. Okay? Any questions? No? Okay, now I will ask Javed sir to come to the podium and explain you how to do all of this. I guess it's quite clear, but still in the Excel.
Here, Sir and Shanu, have you explained you all of the things? Sir explained about the theory behind the qPCR, and Shanu told how we calculate that fold change from the CQ value. So here, that is the file output, how we get it from the qPCR machine. This is looks like, it shows here position, what position you have. Right. This is. Here, this is your time point. If you have, we used here our own data set, and here this is the multiple time point, like yesterday we are using 0 hours, 24 hours or 72, whatever we have. Here, this is our time set. What does it come? Okay. Here, next, this is your condition. What condition you preferred? This is untreated or treated. For example, we used here 0 hours, so we can put it here as this is untreated. But next treatment, whatever we have given, whatever we did, took the data, we took as treated condition. Important is here the CQ value. This is the range of the CQ value. You can see here in sample 0, there was 3 replicate was there. There was 3 replicate was there for actin, and here I mentioned G22, G22 likewise. This is 3 replicate for each of the genes. So further we have to arrange the data for the calculations. Here I arrange the data in a simpler way to calculate the fold change. First, this was the sample name. I aligned the sample, 0 hours, it was 6 sample was there, 0, 24, 48, 72, 96, 120, and this is NT. It was no template. Gene name I put it here. This was three condition, CQ1, CQ2, CQ3. This is the, from each replicate we get the CQ value. First we counted the mean. Parallelly we put that actin for the same. Actin also have each 3 sets, CQ1, CQ2 and CQ3. So for the reference gene as well as actin, we calculate the mean value, as Shanu explained very well in previous slide. Next comes how we put the formula. First formula was, what was that? Exactly, delta CT. First we have to calculate the delta CT. So what we did to calculate the delta CT, if you see here the formula, what I did? Anyone? Exactly. So I reduce, here 2 sample was there, actin and my reference gene on same time point. So for my genes, I reduce that actin. Sorry, the formula is little bit. Wait. Is it okay? So the delta CT came for all of the value. Got it? Here one thing, I used here 3 genes. That was that G22 is the gene 1 for me, and G28 is gene 2, and G38 is the gene 3. So 3 set I had to prepare for a 6 time points. So next what we will do, we have to calculate the delta CT. What we do for that, we used our gene and have to reduce our control sample, right? So here same thing we did here. That is our gene set and minus our control sample.
The treated and the untreated. This is the story here.
So we got it here, delta CT value. What we did next, we have to put in the formula to calculate the fold change. 2 power minus delta CT. We put it here as such formula. It looks like power in bracket 2, that was the, and minus value of delta CT. We just put it that under, the value will come here. So here finally we will get that fold change value. This is as the show in yellow color, this is the fold change value. Here we can plot the graph. Either we have to plot it here in as well as in Excel, or we can export in any of that static software that Shanu told earlier. Here further we have to go for the statics analysis. Here we took the only one value. So if you have to calculate the statics, we have to use at least 3 values to calculate the mean, standard deviation, standard error, and any of the statics related. Same value have to calculate from the same time to second time set, next value have to calculate from third time set. So we get the 3 value here, and we go for the mean, further we'll go for statics analysis. So this is finally you got the fold change. If you have any doubt, you can ask. Yes, sir.
What if expression of our target gene is lower than expression of reference?
Okay. How we got it that whether, how we decide whether gene is upregulated or gene is downregulated?
So if the treatment the expression of gene in the treatment, if it is lower than the reference gene, and if there will be no any expression in untreated gene, untreated so if the treatment expression of the gene is lower than the reference expression, and if there will be no any expression of the gene in untreated, so even though we can consider it as a downregulated.
Actually, all of the gene set have to express, but how much they are expressing, we got to know about it. We select it as a standard actin or whatever housekeeping gene. Based on that, we count that relative. If housekeeping gene expression is going to 1, but our reference gene is going to 2, either 0.5. So based on the comparison, we can say that is downregulated or this is upregulated.
Housekeeping and reference gene is same.
Huh?
So
Housekeeping gene, irrespective of you have to give the treatment or not, we are going to express. Okay, even if it is uneducated, it is going to express. And when you will do the calculation, it will come as the normalized, because when we are doing the calculation, we are actually normalizing the CQ value. So it's going to come as 1 only. So that will be your threshold value. Okay, above this, it will be your upregulation, and below this, it is going to be your downregulation.
so housekeeping genes value will be the same, whether it is treated or not. Right?
Yeah, in all of the conditions.
But the problem is there, our gene of interest, if it is not expressing in untreated condition, and if it is expressing in treated condition, but it is lower than the target means reference gene. So will it be considered as downregulated?
It is going to be considered as
Sure. Yes.
Even if it is expressing in the treatment.
You got the expression everywhere, but how much expression was there that you have to calculate? What you say,
Like if CD value, if the CD value is coming 40, if you consider it 0.
there will be.
From that, how much it is so let's give, I'm giving one example. Like expression of reference gene is 20, CD value is coming 20, and the expression of treated gene means the gene in the treatment is coming 70. Okay, and the basal value, what the same, there will be basal expression, and if it is sensed by the machine, and if it is coming 7. So 7 is also lower than the reference. 17 is also lower than the reference level.
There is a range of CQ value. What we generally consider from 18 to 30.
you said, no? So it is 19 and 18.5.
All genes are going to upregulate.
No, no, it is lower than the reference gene.
You're talking about fold change, you're talking about a CQ value. If you're talking about CQ value, that's all genes are going to upregulate. We have a standard range, but we consider it from literature and from experimental design that this is 18 to 28 is the most probable value that's come into the expression level. This is the CQ value. If CQ value is high, that expression is low. But if CQ values comes lower, like 22, 20, then 18 is comes lower, that it means the gene is expressed high.
Even if the reference the CQ value of reference.
Yeah, lower CQ value, higher be the expression.
But in the early stage, ma'am, if we will go with the formula, it comes.
No, one minute. This is your non-directly both your control sample and treated sample with capital. Yes. It is not
The point of comparison is to inoculated and non-inoculated, not with the reference.
Reference have to use only to normalize the data.
Normalizing both.
That's it. Only for normalization. Anyone else, please? So you got to know how to run the PCR, how to analyze the data. Any doubt from here? You? No, we got the data from the machine. Actually, it has some missing value, and you know how it would be missed because of our handling issues. And this, huh? Okay, okay. I'll show you the data. Whatever we got, I can. Huh? The same data. The same data we got it. Just I'll. See here, this is the data we got from our qPCR machine. Most of the things are missing here, so how we will go for the analysis? The data we get the same data. I shown you that experimental data. This is same we will get it, should get it here, but we could not. So this is our data. How I fill the gap. That's why I didn't calculate it.
That okay, in this well, this is the condition I have provided, and according to time points, you can do the editing. So, if we didn't do that, I have to go back to my normal data, which I had in my, because I provided the cDNA from my sample. I have to go back to my data and, label the wells, and then I have to do the analysis, that will take time. And I guess we got this data in the morning only. So, because the machine took time for running. Took almost 2 hours. I was there, but I couldn't take the data yesterday. I took the data in the morning. So, one more thing. Sometimes to label the wells before you read the thermocycle reaction, before you do that, it's better you label the wells before it's in hand. But in some machines like Roche, like Cycler, you have to do that after you get your CQ values. Okay, but there is a plus point in Roche. You can actually, do the absolute and relative according to what you require. But in this bioreactor, I don't know if there was an option. It was almost relative. So, yeah.
So what is next?
I guess feedback. We all require that.
Yeah. Thank you, one and all. Please do it.
Assemble there and have some refreshment. If there's any queries, yeah.
Let's have the tea break.
the profiling. Okay, the word profiling means you have to sequence the sample using a kind of marker, what is called as metabarcoding. What was the marker? Was 16S ribosomal rRNA. There is a specific marker, V3 and V4. So we use that as a marker to profile the microbial content of that microbiome. So that is a downstream work. It involves usually two parts. One is, three parts I would say. You start from wet lab, go to in silico, and come back to again wet lab for validation. Remember, always starting from wet lab. Sometimes you start from even in silico first to identify the markers. So it is in silico to wet lab to in silico to wet lab. So it is always shuttling between these two, right? But the most important thing is the biological sample preparation, right? All of you may be knowing how to do that, but still, just to give a refreshment about those knowledge, we'll take it. The microbiological sample preparation for genome sequencing is a very, very critical step in obtaining high quality genomic data from microbial sample, right? Always remember whether your sample is really representing the system which you are handling. This is one of the biggest challenge, whether it is a rhizosphere microbiome or it is a phyllosphere microbiome. The sample should represent the entire system in which you are working. For example, you are collecting a sample for phyllosphere microbiome, right? You are approaching a plant which is around 45 days old. The question is which leaf you will sample it, correct? We know that the microbiome composition varies from leaf to leaf. The leaf which is broader, which is matured, which stayed for quite a long time in the environment, it might be harboring more microbiome as compared to the one which is just emerging in the top. So whether it is a representative of the entire plant, always keep this point in your mind. So the best way forward is you collect a bit of leaf that is representing the entire plant. It is always a pooled sample you have to collect. Okay? That is one part of the story. And the proper sample preparation or proper preparation ensures that the resulting genomic data is accurate, complete. The complete in the sense whether it is really representing the entire plant or entire system. So I talked about the individual plant. And what about the neighboring plant? Whether the individual plant what you are sampling is really representing the entire field. Another challenge, correct? So that adds a bit more complexity now. You have to go for one more plant, right? Now the question arises, the second plant also is representing the entire field. Then perhaps you may add one more plant as well. So the number increases. So if you look at the papers which is being published, the first question the reviewer asks whether you have a replicated sample that is put into the downstream analysis. Remember, never ever do this microbiome analysis with only one sample. There must be at least 2 biological replica. I am telling at least 2. Any number above 2 is desirable. Okay? That is a biological replica. And what about technical replication? You need to have a technical replication as well. So 2 technical replication and 2 biological replication. That means for each treatment, eventually you are going to handle at least 4 samples, right? This is in terms of number of units what you are going to put into the downstream work, okay? Yeah, just have a reading. The proper preparation ensures that the result of the genomic data also includes the so-called microbial metagenomic data. I think this is explained in the manual. Please go through that. Is accurate, complete, and representative of the microbial community in the original sample. Always remember, whatever you are making the interpretation, whatever data you are collecting and making an interpretation, that should be reproducible, right? The reproducibility is a challenge very often in the case of microbial metagenomic analysis, right? This is a question the reviewer or the readers are going to ask. How reproducible the results are. If I repeat the results, are we going to get the same microbial count in the second time? And how confident you are in generating the same data set again. So these are all some of the issues we need to address, typically with the number of samples only. Okay? Yeah, the first step in the microbial sample preparation is to collect the sample. The sample can come from a variety of sources. It could be a bulk soil, it could be a rhizosphere soil, it could be it could be a kind of a rhizoplane, or any biological tissue. It could be a stem, endogenous region, phylloplane, phyllosphere, flower, fruit, and it can be anything. And once collected, the sample is transported to the laboratory and processed immediately. Always remember, this kind of work, there is the continuous flow of activity. Many times, okay, you collect the sample and you feel, okay, my job is done. No, actually the job is initiated. Because any delay in the processing of the sample, remember, you are going to generate a data which is not representing the original microbiome composition in the natural field. The moment the leaf is collected, and all sort of metabolic activities are changed because the plant is not in connection with the nature, correct? The leaf metabolism is changing, and what change it is happening there? It is going towards a death phase, isn't it? Sooner or later that leaf will dry and it will dry off. That means the leaf is not in the original state. So this is one of the reasons why I am suggesting whenever the sample is collected, it needs to be processed as soon as possible, right? Preferably in the same day, correct? So that is why it is to prevent any changes in the microbial community composition, correct? There are microbial members, they are very sensitive. They may not even survive when the leaf is removed from the main plant. The counts will be, it may die off. Of course, metagenome data may pull out that, but when you go for a validation through culturomic tools, you get all kind of discrepancies, correct? So the timing and the processing should be, the collection and processing should be as close as possible, okay? Of course, I'm not getting there. The sample need to be processed. Rymaker was repeatedly telling that it is the same sample is analyzed using different approaches, correct? A portion of the sample goes for the so-called meta-barcoding if it is a microbiome profiling. A bit of a sample is going for culturomics through a plate count or plate assay. A third portion is going for maybe imaging, right, using different kind of microscopic analysis. Right, yesterday one of the groups were talking about the scanning electron microscopic images of rice leaf. It's all done in the same sample, right? Maybe the fourth sample may go for a long-term preservation as well, right? So this is a typical workflow. When the sample is collected, keep all these things in your mind. A portion of the sample goes into a metagenome or meta-barcoding analysis. Yeah, another portion goes for culturomic or microbiological methods, what is called as a culture-dependent methods. Third one goes for maybe a microscopic analysis, and fourth one maybe for a long-term preservation. And you can add up. Suppose if you are doing metatranscriptomics, maybe another sample can be going for metatranscriptomics, metaproteomics, and even metabolomics. So what I'm trying to tell, it all depends upon your biological question in your mind and what kind of analysis you are going to do, and some integration of all the data is going to answer the biological question, what you have in your mind. Everything should be considered in the day one, okay? And of course, the DNA need to be isolated, and I'm not going to elaborate further because we have already discussed about that in the practical. You did a DNA isolation also, and variety of methods are there. You have a lab-grown method is there, you have a kit method is there. I can share our experience, what we are doing in our lab. We never depend on one particular method of DNA isolation for downstream work. We always integrate at least 2 different methods. Method number 1 is invariably a kit-based method because they are likely to give you high quality, high molecular weight DNA for downstream analysis. And also, a portion of the sample goes for a DNA isolation by adopting a lab-grown method, right? And 2 independent methods are deployed here to isolate the genomic DNA. right? And the two DNAs, whatever coming from the same sample, is going up to perhaps polymerase chain reaction. Amplicons are generated, and amplicons are mixed end of the experiment. That goes for sequencing, right? Because that is how the workflow we adopt in our lab. And of course, the DNA isolation has got its own, issues, like it should be a high molecular weight and highly pure DNA. I think the NEO team has already conveyed to you what should be done. Okay, I'll skip this DNA isolation. Then after DNA isolation, it goes into the sequencing pipeline using machines. The choice of the machine depends upon your type of objectives. It could be a short read sequencing or long read sequencing. It could be even metagenomic sequencing by shotgun. A variety of tools are there. Okay, and finally, of course, the reads are coming to you, and the reads are, of course, it was already handled very elegantly by Guillermo and Luisa. How to curate that sequences. The ends are trimmed, poor quality reads are removed, and the high quality base calls are taken on board for any kind of further interpretation, right? So that has to follow certain pipelines, and most of the softwares and programs are already communicated to you. Please follow that, right? Of course, this is a very important point. What I want to conclude here in the first part of the three different approaches, what we're talking here, the microbiological sample preparation for genome sequencing and metagenome sequencing is one of the most important crucial step in obtaining high quality genomic data. Remember, if that is wrong, and the mistake would amplify, right? Remember, mistakes will amplify with the time and money, and time is money. All right? So keep this point. Your sample preparation should be as precise as possible. If you make any compromise on that for one or other reasons, if at all any error, knowingly or unknowingly, if it is happening there, that error will multiply. Okay? So don't yield to that, and be careful with that. Of course, these are all the sum up: proper sample collection, DNA extraction, fragmentation, library preparation, sequencing, are all necessary to ensure the resulting genomic data is accurate, complete, and representative of the microbial community in the original sample. Coming back to the same statement what we started with. Now, I brought two case studies. One is not case studies, two main biological material what all of us are comfortable with. One is a bacterial sample preparation, and another one is a fungal sample preparation. I think those who are handling bacterial pathogens or microbiomes or fungi, this must be boring to you, but those who are not from that side, I think it's an information for you. How to prepare a bacterial sample for sequencing, whether it is a single pure bacteria for whole genome or it is a community bacteria for metagenome, right? The story is same only. Remember, if it is a pure culture of bacteria which is subjected to some kind of identification by a marker gene paste or the whole genome, the purity of the culture is very important. I always tell 2 points. One is microbiological purity, and another one is genetic purity. You cannot compromise on these two. Microbiological purity means the bacteria should be free from all other bacterial species. Okay? Genetic purity means it should represent that particular strain of bacteria which you are interested in. Remember, same species of two different, same species, but they are two different isolates. They may coexist in the environment. Correct? Especially in the case of, we have seen in our own data sets. You have a variety of different types of Pantoia agglomerans living on the leaf. Can we say that all of them are the same species? No. They are different strains. I would say different isolates that qualify to become a strain when you identify some trait associated with that. I would say they are different isolates, right? And in all, in our own opinion, two species of Pantoia agglomerans, we have seen they have a different functional potential. So meaning what? So they are not same. There are differences. So you have to segregate them. that is what I called a genetic purity. All right, remember microbiological purity is first. It's quite easy to do. Mostly it is through morphotyping. Two different bacterial species on the same plate, you can segregate them just by visual observation, right? For example, firmicutes and pseudomonas, you can very easily differentiate. Bacillus, I think most of you, those who are working with bacillus, you know how the bacillus looks like. You can segregate them. There is no problems on that. But if two different pseudomonas coexisting on the same plate, that raises lots of doubts, right? So to circumvent those confusions, only in our lab we have developed a very elegant technique. Can we decorate the bacterial colonies by adding some supplements in the medium? I think one of the papers we published in Environmental Microbiome, I do not know how many of you noticed, the petri plate was, the colonies were looking blood red in color, some of the colonies. It's no magic there. We have added some chemical, 2,3,5-triphenyl tetrasodium in the medium. So what it does, it gets converted into formazole, insoluble salt, and that is accumulated in the bacterial cytoplasm, and it decorates the bacteria. That would help you to some extent. I won't say that it is going to help you all the way down up to segregating two isolates, but certainly it is going to segregate different species of a particular genus, correct? So the colonies will look different. So that is assisting you to take out the different species under the same genus. Okay, so this morphotyping with assistance from a dye, I would call a dye-assisted, redox dye-assisted morphotyping. This is one of the very elegant technique that would help you to do the microbiological purity of your sample, right? Now you are entering into another stage. Okay, I have isolated the same species, but are they same? If there are too many colonies are looking alike in the plate, whether all of them are all of them are same species with the same functional potential, or they have some finer differences. That's what, remember, Yost was repeatedly talking about, look at the strain level. I do not know how many of you picked that point. The real value is in their strain level, right? So what is the way out? And here you cannot do the morphotyping, very difficult because they look alike, isn't it? So what is the way out? Here comes the DNA fingerprinting techniques, right? So one of the very elegant DNA fingerprinting technique developed for bacteria is what is called as REP-PCR. There are different variants are there, but the one which is very popular is a box PCR. And if you use box PCR fingerprinting, that would reveal finer differences between very two closely related bacterial species, right? Just by counting the number of amplicons, presence or absence of certain amplicons, you can very easily segregate very, very closely related strains or isolates of the same species. You have to do it, okay? So the main point is microbiological purity is assisted by redox dye, what we are doing, and you can develop your own method to do that. Maybe different kind of approaches are there, right? And the genetic purity you can use or you can do with the help of a DNA fingerprinting technique. Yeah, please go through that, some of the points. Of course, the single colony isolation is even today, I would say, is going to be forever, is one of the finest technique for purification of bacteria. Never ever underestimate that, right? You tell me how many of you can really bring a single colonies in third and fourth quarter of a quadrant stick? Yeah, if you are doing it, I'm very grateful, but many times people fail, right? Third and fourth quarter will be still having the trails of lines without any isolated colonies, right? So the best method, I always tell the third and fourth quarter should give you at least 20 independent colonies, at least. If you can do that, and you are well set for handling bacteria, right? And how to do that? Anybody? It's very, very basic. Very simple. The loop what you are using, after every streak, you re-sterilize it. Very, very simple. No magic here. Okay? You do that, you will get third and fourth quarter. started, individual isolates. Single colony isolation is one of the very elegant technique, and remember that is having a far-reaching implications if you are not doing it properly. Imagine you are taking a streak of bacteria in the third and fourth, you are not able to get a single colonies, or one stray colony little away from the streak. Okay, you are picking it up, but there may be a possibility it is a kind of a microbial artifact, it's a contamination. Then everything will go wrong, right? So never ever compromise on that. You should get at least 20 independent colonies, they look alike, right? And indicating that what you streaked in the first one is the one what you are seeing in the third and fourth. So very important point, okay? Then the next work is once you microbiologically purified and genetically also purified by using DNA fingerprinting technique, the next step comes, you have to mass multiply it. And again, the question is, in mass multiplication step, what are all the checks and are there so that other contaminants are not appearing there? Maybe there may be a possibility something else may grow by, you have to take care of all those finer details so that what you inoculate is the one what you really want, and what is multiplying is the one what you really inoculated, and finally what you got as a biological material is the one what you really wanted to sequence or do the microbial analysis or metagenome analysis. Every point you need to be very, very careful. If you make any mistake somewhere, the entire things will collapse. Okay, be careful on that. So of course, broth culture is a very elegant technique for bacteria. You can grow overnight and reach the saturation point or mid-log phase. Mid-log phase is one where all the bacterial cells are in the active metabolic state. What is active metabolic state? They are in the dividing state, right? So that is a point you have to stop it. Why you have to stop it there? Because too much of growth is actually a problem, because they accumulate a lot of unwanted metabolites. Those metabolites maybe sometime may interfere in your downstream analysis, right? So the best way forward is the day one, if you decide to go for genome sequencing of a bacteria, we'll come to metagenomic a little later. The best way forward is you optimize the growth conditions and optimize the time you have to stop the bacterial growth. That stopping time is something which is going to give you metabolically active bacterial cells. This is popularly called as mid-log phase cultures. Okay, there is no single time point for most of the bacteria. You have to standardize it, right? So you have to optimize it. You have to in a 2-hour interval. You have to take out the sample. You have to go for OD measurements, and you have to go plate it out. And this generation curve analysis or the growth curve analysis, you have to do it. So that gives you an idea like, okay, this is a time after 24 hours of my inoculation, my bacterial cells are ready for stopping and ready for the downstream work. Remember, not only for metagenome or genome sequencing work, but also for functional assays, right? You want metabolically active cells for any kind of functional assays. If you are taking a cell which is in the dying phase or death phase, certainly they may not do their job properly, correct? The death phase already started and cells are aged, right? So we need something which is in the very dynamic state. So mid-log phase is the one which is always useful for any kind of in-depth, in-depth sequencing work and also for functional analysis. Okay. Excuse me. So grow the culture to the stationary phase. Before stationary phase, you stop it and take out the culture and harvest the bacteria. Harvesting means a very jargon here. That means you stop the growth and separate the so-called culture filtrate or the bacterial metabolites from the cells. That is usually by a centrifugation. So the cells are pelleted, supernatant is removed, go for 1 or 2 rounds of washing, preferably using a saline 0.8% sodium chloride or phosphate buffered saline, and your cells are free from any kind of metabolites which came out of the bacterial cell, and the cells are ready for any downstream work. Okay, and of course DNA extraction, quality control, we already talked about it, and I'll just skip it. Library preparation, sequencing, that is a flow. You have been already popular, in the sense quite aware of that. It has been told more than several times here already, right? So in conclusion, purification of bacterial colony is a very crucial step in preparing the biological sample for genome sequencing or metagenome sequencing. This process involves growing the bacterial cultures, harvesting the cells, extracting the DNA, assessing the quality of the DNA, and of course, the DNA is subjected for fragmentation, library preparation, and sequencing, right? This is with respect to a pure culture of the bacteria. You may wonder how to handle a metagenome sample, right? Metagenome sample, no different from that. The only difference here is you are not going to subject the sample to culturing technique, rather you are going to directly extract the bacterial community from the environment. Okay, how it is done? Quite easy. Rhizosphere, you have already witnessed that day, right? And this is thoroughly explained in the manual as well. For phyllosphere, there are variety of techniques are there. First and foremost technique is extraction by washing. You have to wash it. You have to wash the cells from the leaf, right? Washing is, what do you mean by washing? You are going to dip the leaf, which is representing the entire plant, maybe representing the entire field as well, right? And you are going to put the buffer. Most often the buffer is phosphate buffered saline. And to reduce the surface tension, you have to add a 2N20 also there. So that gives you a kind of option for the cells to get removed from the the leaf. Remember, bacteria is not living as a kind of a passive life there. Most of the bacteria which is adapted in the plant associated niche, they have a very active life there, right? And most often, at least in the phyllosphere part, I can say that they live in a community there. This is what is called as bacterial aggregates. The bacterial aggregates, they never live in a solitary cells on the leaf, right? Because the solitary cell on the leaf is something the cell may die because of the vagaries of weather. So they always live in aggregates. Aggregation, I think most of you might have heard about the bacterial biofilm aggregates, because they cooperatively live there so that the cells are protected from the so-called desiccation and other vagaries of weather, right? I'm sure in rhizosphere also the story is same, right? So you have to detach the cells from the surface. For that, perhaps the 2N20 might help you. And repeated washing. Usually the washing is done, it is called a sequential washing. 5 to 6 times you have to do that, right? So the ratio, usually it is 1:10 ratio. For example, you take 1 gram of leaf and 1:50 ratio, you can add 50 ml of buffer. And you agitate it for about 15 to 20 minutes, preferably 30 minutes, right? You collect the supernatant and add another load of the same buffer and keep on collecting it 5 to 6 times. So at the end of the experiment, you end up in around 300 ml of buffer, right? This 300 ml of buffer is something which repeatedly you harvested the bacterial cells into the broth, into the suspension. So now this 300 ml, whatever you harvested or whatever you extracted, need to be pelleted down by centrifugation, and the pellet is finally suspended in 1 ml, right? That 1 ml would carry most of the microbial cells you wanted to isolate, right? Now the question is, why 5 times? Why not 2 times? Or why not 10 times? Right? You do it yourself, you will understand why 5 times, because on 5th and 6th washing, you start getting some chlorophylls started peeling off. Right? That is an indication that you have taken everything. So what is there is the plant started leaving some of its material to the extraction. So you can stop that comfortably, right? So now you have 1 ml microbiome, total phyllosphere. And I'm sure the story is same for even a rhizosphere also. You can go for repeated washing, right? And this 1 ml, again, segregated into the 4 things what I already told, right? Something can go for metabarcoding or metagenomics, something can go for plating out in the serial dilution plating, something can go for even imaging, something can go for even preservation, right? So that is all about the biological sample preparation for bacteria. Now, a couple of more slides on fungal purification. Many of you may in future handle a microbiome analysis, right? So that is slightly different from microbiome analysis. The microbiome analysis, although it is conveying the entire microbial community is there, but in reality, people address only the bacterial diversity there by and large. And if it is fungi is your target, then probably you may call it as a microbiome analysis. using ITS markers that we have already discussed in the class. So here, story is again same. There is no big difference. The processing is exactly the same. No difference. The only difference comes when at the sequencing part, the marker what you are going to use is different from the what you are using for bacteria. What I'm trying to tell here is extraction is same. You can use different approaches for exploring different microbial domains, right? If you use V3, V4 marker, you are addressing the bacterial diversity analysis there. If you are using ITS, perhaps you are addressing the fungi, right? For oomycetes, for protists, for other microfauna also, you can deploy different kind of markers to analyze all microbial life which is there in the environmental samples. Okay, so with respect to fungi, the challenges are slightly different because fungi are, they are having a very thick cell wall. Most often it is the chitin. And the chitin is something which is a very strong polymer, so very difficult to breach this strong polymer. You need to have a different kind of strategies. Please have a reading here. The fungi has got a very tough cell wall that can be resistant to lysis. It is true because many times people fail in isolating a fungal DNA. You have to go for a physical and chemical force, right? Sometimes we use a finely powdered glass as an abrasive material to really crush the fungal cell wall so that the inner contents are coming out. That is recommended also, and you can use even a liquid nitrogen if you can do that. So the main point here is to bring out the fungal DNA from the inner region is always a big challenge, right? But of course, we have an excellent fungal DNA isolation. The kits is available, and you can develop one in your lab as well. Okay, so the thing is, DNA isolation part, I will skip it because you will be taking the help of some kits, or you develop your own laboratory method for isolation of fungal DNA. But the most important point is, again, I'm coming back to the same story, the microbiological purity and genetic purity, right? Bacteria, from that perspective, they are quite easy and quite comfortable because they are single cell organisms and they develop a single colony. Single colonies are progenies of the same genetic makeup. So there is no difference in the progenies if you are picking the same colony. But fungi is quite different because isolating a fungi that to the genetically pure form is always a challenge. You know what is the best technique to ensure the fungal colony what you are taking or fungi what you are taking is genetically pure? Anybody? It's a hyperlative isolation. Very old technique, but still, much like the quadrant stick, still it is ruling the so-called fungal purification, right? Hyperlative method is the best method. You can go for a water agar and place the fungi on top of that. In water agar, they grow faster than the normal media, and the leading hyphal tip you see under microscope, mark it, and just using a cork borer to take it out and put it in another plate, the best method. No other method can replace this method, okay? Hyperlative isolation, all right? So use that technique, and of course we cannot give an opportunity for you to practice that. It's quite easy to do. You can do it in your lab. Very cheaper method of purifying the fungi, right? So these are all some of the steps what I wanted to tell you, because fungi are ubiquitous. They are everywhere, right? Bacteria. So sterilization assumes its own importance here. Ensure that all your experimental tools are properly sterilized. Your flow cabinets are properly maintained. There are no carryover contamination from the previous work. Filters are clean, and these are all some of the housekeeping thing you are supposed to do before addressing this fungal isolation, right? And hyperlative method, you follow that. Once it is removed from the master plate, right? Put it in the new plate. Try to grow a colony into an adult colony. From there, you take the biomass. And here comes the next step of mass multiplying the fungus. It is usually done much like bacteria in a broth only, right? You grow them in a broth, and only thing is the broth composition varies. For bacteria, normally we go for TSA, that is tryptic soy broth, or you can go for a nutrient broth or any high nutrient-rich medium you can use for bacteria. For fungi, you can use either Sapex agar, you can Sapex broth, or you can go for potato dextrose broth. There are a variety of media which is available. You can use, choice is yours, right? Grow them, and always grow them in a agitation mode. Never grow the fungi in a static mode, right? So if you are growing fungi in a static mode, they form a thick mycelial mat on top of that, and you will find it so difficult to do the downstream work, especially the DNA isolation, and that's going to be a big challenge, right? You have seen one of the slides, yours was demonstrating how the fungi is forming a sclerotia. I think the video was a very nice video. So they really form a kind of a thick mycelial mass, and they are very difficult to segregate them, and they resist most of the attack, what do you call, lysis attack. So better you grow in agitation mode. In agitation mode, I do not know how many have an experience on this growing fungus in agitation mode. You get a fungal balls, right? So how many of you have experienced it, right? Yeah, you get a tiny, small bubbles kind of fungal. They are the ideal material for DNA isolation, right? So take them on board, and of course, the same story. A portion can go for metagenome, something can go for long-term preservation. You can image it. You can go for any kind of omics tools that is in the pipeline. Okay, so that's all I wanted to just tell, because it's a very basic thing, but very important thing for your downstream work. Okay, so let us take some questions if you have anything. Yeah. Put on the lights, yeah, please. Anybody? No? Okay. I expected a lot more questions from you, but no questions are coming. I would take it as a compliment for my lecture. Okay. Thank you. So the next speaker is already here, and of course I'll be here. You can ask the question throughout the day, the coming day, 2 more days also. There's no problems. Okay. Thank you. Yeah. metagenomics of plant, concepts and application. This lecture will be delivered by Arpita Ghosh. Arpita Ghosh, currently an assistant director, bioinformatics, working with the Eurofins Private India Limited in Bangalore. And Arpita has got a very long experience in bioinformatics. And she has been one of the very important technical support for this company, Eurofins, and she has been sitting IRA for a long time. And in the past also, Arpita has delivered excellent lectures for the trainees. And I was having no hesitation to invite her for this program as well. Right? Arpita, the floor is yours.
Thank you.
And the students are PhDs and MScs from IRA, and a couple of them are from IITs. Then we have staff, Dr. Ramakrishnan, Dr. Govinda Swami. We have many other people in the audience. They are keen to learn the metagenomics. Right? Last 7 days we had a program with experts from NIO, that is Netherlands Institute of Ecology, and Leiden University. I think 4 experts were here. They were exposed to some sort of bioinformatic tools and the techniques. Let us take the message from industry perspective, what they are going to tell us, right? And try to connect all the dots. Okay? Yeah, best wishes to you. Madam, to you.
Thank you. Thank you. Thank you for the lovely introduction. So I will start with the basic and then I will take you to the bioinformatics, what are the different approaches and how we can apply them and what are the outcomes we can get from there.
If it is not working, you can use this one as well. Okay.
Yeah, I'm fine with this.
Okay.
So I will first talk about Eurofins. So Eurofins is a global company wherein we are in various different countries. We are totally in 50 countries and we are somewhere around 900 plus labs across the globe. We are working on genomics as well as other things like food testing, environmental testing, your other microbiomes, the other analytical sites, all sorts of testing are done by Eurofins. I am representing mainly the Eurofins Genomics India. Eurofins Genomics is another thing which we are operating from these countries and we are in India in Bangalore. We are also having a small operation in Hyderabad with CCMB. What is different in Eurofins is that we have range of services starting from oligos to your next generation sequencing, microarray and other services. So we have food testing in genomics, we have your oligo, all sorts of oligos, gene synthesis. We have an in-house gene synthesis facility which is coming up. We have agri-genomics, specifically the genotyping arrays. We have developed few of the techniques which are only with Eurofins Genomics India, where we are doing lots of genotyping. We are doing lots of big microbiome projects with government, where we are doing lots of human microbiomes and lots of wastewater surveillance studies. We have done it in COVID times. We were part of the government to identify the COVID strains across the country, how the COVID mutations are moving from the wastewater management. Then I will start with metagenomic approaches. And I will give you a brief how the entire overview works and then we'll go in the detail of the analysis. So already, as I believe you have already learned what is metagenomics and what it is. So in brief, metagenomics is having different techniques to identify what is our aim, what is our objective we need to know to decide which is the service we need to opt for. Like for example, metagenomics can be done in different ways, like that is shotgun metagenomics and amplicon based metagenomics. Now when we say amplicon based metagenomics, that comes 16S, ITS. There are specific markers which can be done for a specific type of microorganism which we are trying to detect. When we are doing an amplicon based microarray, what we do is that we we decide a marker, gene marker like 16S or ITS. We take the marker, we assign a barcode along with that so that we can differentiate between the samples, and then we sequence. So we have the Illumina adapter which is there, and we have the primer sequence ahead of that, and we have the indices which we are showing you in different colors, 4 colors we have shown here. So what happens in this case, we don't have to do a library preparation. That's the reason when we see 16S or ITS, that's pretty cheap or pretty low on cost than your other shotgun metagenomics. How we reduce the price is that we prepare the primers with the adapter sequences of Illumina, and then we sequence out the amplicon itself. Now when we do this particular method, we are just doing the indices steps which comes from the Illumina indices. We can do NEB also we can use. We are just adding their indices. We can develop our own custom primers. Like for example, you want to do, we were talking about nitrogen fixation. If you want to identify nitrogen fixation bacteria, we have specific marker genes for that as well. For example, if you want to do some nematodes in the soil sample, there are specific markers for nematodes as well. So we can take those markers, design it with the adapter and sequence it. So this is a very economical step to identify the abundance. In this, we know what is the abundance, like what are the organisms, how they are going to come, like what are the percentage of organism. If you want to do only the abundance part of it, then we go with amplicon-based approaches. But if we want to see what is happening at a gene level, what is the functional abundance, what are the functional changes, or what are the functional categories which are happening, then we have to go with shotgun metagenomics. We will talk about those things. So there are different approaches, as I said. We can take any sorts of samples, whether it is water, soil, human gut samples, fish, anything, insect, anything can be used for microbiome. Now, when we are doing this, we have different techniques like 16S is for specifically for amplicon based, ITS is amplicon based. We have shotgun metagenomics and we have metatranscriptomics. Now, for metagenomics, when we are trying to do functional abundance, we are trying to identify what is happening at a gene level. We are trying to identify what are the microorganisms across, like whether it is eukaryotes, fungus, bacteria, viruses, all together if we want to do it, we go with metagenomics. And metatranscriptomics, when I want to know what is happening at the pathway level, the gene expressions. What is happening to the gene expression, where the gene expressions are going up and down. So if we want to do that, then we go for metatranscriptomics. Now metatranscriptomics is based on the RNA-based test, where we are trying to do the expressions of genes. We need to understand that RNA from these type of samples are pretty difficult to take out, because you have a soil sample which will also have your plants, microorganisms, your soil itself will be having some inhibitors to take out the RNA. So this particular application, RNA, is little difficult to do the extraction part of it, but there are specialized kits, specialized applications which can be used to extract the RNAs, and the RNAs will be in a very small proportion, right? So if you are trying to identify the abundance of a particular sample and you opt for metatranscriptomics to do that, then you will go wrong on your particular results outcome. Why? Because your abundance will not come completely. That will only talk about your RNA part of it, which is based on your expressions. Now, in that, if I try to identify the bacterial abundance, I might not get the full picture of my abundance of that particular sample. So we need to know why we are using these different services and why these applications are so important. Now, if I am trying to do an abundance, 16S or ITS or a particular gene-based amplicon is helpful. Why? Because I'm trying to capture a particular region of all the set of microorganisms which are present in that particular sample. When I'm reducing my search criteria to a smaller part, then what happens is that you are able to fetch out a similar uniformity from all the particular organisms which are present there. When I do that, my abundance is better than having a total DNA which is there and trying to identify what is there in that. Right? Then I will miss out the rare ones, I will miss out the smaller organisms or smaller microbes which are there. Why? Because I have other stuffs also which are there. Now, but in viruses, what is it? Now, after COVID, many people are doing virome analysis, vironomics, which is coming up. In plants or in soil, we are trying to identify what are the viruses as well which are present. So how do we do that? We don't have a marker there. There we don't have any option, then going with shotgun metagenomics. So in shotgun metagenomics, we take the entire DNA content of the particular sample and then go ahead and sequence the entire DNA with a WGS approach, and then we see the actual abundance, and from that abundance we pick up only the viral part using different custom databases. There are specific viral databases, RBDB is one of them. You have RefSeq virus databases. So we pick up these virus databases. We have also developed a custom viral database, and we pick up these viral databases and hit against that and take out the taxonomic abundance of viruses. Right? In those cases, we have to go with shotgun because we don't have a set markers in case of viruses. But for bacteria, archaeas, for funguses, we have set markers, the gene markers from where we can take out the abundance. So we need to understand the importance of these services and these applications so that we know which application goes with which type of objective or which type of aim. This is very important, otherwise what happens is that you will get an outcome, no doubt about it, but your outcomes will not be good, and then the reviewer will be questioning you and you will not be able to answer it, you will not be able to justify your things. So going in more details, so in 16S, specifically I've taken up this example because many of you people must be doing bacterial 16S. Now in 16S, if it's a pure culture, we know that we go with Sanger and identify that particular thing. We are doing when it is a mixed culture, where we have mixed sort of stuffs, and we are also capturing an uncultured bacteria as well. Now there are different variable regions which can be captured. V3, V4 is the most common one which people capture. MySeq is a 2 into 300 chemistry, and this V3, V4 region is somewhere around 450. So it's easy for us to take the amplicon and sequence it. But there is a technology of long read, which is nanopore or PacBio, which can sequence the entire 16S gene. So now what is the difference between a short read and a long read? When we are talking about short read sequencing, we can do short reads. We will have to have an overlapping region in the amplicon for sequencing it so that when we do analysis, we can stitch the reads together. Right? Now in this, we are again making our search criteria very small. Right? We are just taking the V3, V4 region. When we are doing long read technology, we are taking the entire gene to sequence it. Your resolution of your identification will increase when we do long read technology. Right? So we can use nanopore because PacBio is on an expensive side, nanopore is on a lesser side, and nanopore is good for this type of amplicon sequencing when you want to have a huge resolution and like you want to know that in depth what are your rare ones, what are your top ones, abundant, what is it, then long read is very nice. But if you want to do a screening study, you have huge samples, you want to do a screening study, V3, V4 is a good one because we are just making our subset very small and trying to fetch out whatever is present in the particular abundance. What are the bacteria and what percentages they are present and how they are present. So there are two different technologies or two different approaches which we can take in 16S amplicon. One is your long read and one is your short reads. These are the different metagenomics data which is available in EBI. You can go and you can search. Here you will get all the microorganisms which are present in particular sample types, like whether it is soil sample, whether it is water sample, from which
population, how much it is present and how it is, the information is already present in EBI database. Coming to the application part of it, in plants, microbiome can be used in various different applications, whether it is biofertilizer. We have recently done one project for biofertilizer where we are doing soil microbiome, identifying a good growing plant against a poor or a non-growing plant. What is the difference in the microbiomes? We can make biofertilizers using these microbiome information. So these are how we can commercialize our research into the field. Then we have medicines and antibiotics. Antibiotic resistance, there are huge projects of governments which are going on as an initiative, that AMR identifications. I will talk about AMR, how can it be used or how it has been used. Then novel microbes. Novel microbes means new microbes, obviously because we cannot culture certain microbes. We cannot identify with the traditional methods these microbes. So your NGS can give you a platform to identify novel microbes from a particular sample type. But yes, there are drawbacks in this as well, like because we are dependent on databases like Silva or Green Gene, which are your Bibles for the databases from where we are picking up the annotation of the microorganisms. Some of the microorganisms in that also are written as unclassified, but no names are given. So there we will be having an unclassified bacteria, but we don't know which bacteria it is. Sometimes it is like that the top hit is your uncultured bacteria. So these are certain drawbacks on this platform also. But then still we can identify novel ones, we can identify the rare ones, we can identify the top abundant ones, and so on. Then plant diseases is another field where, like for example, viral infection to a plant, if we are not able to identify what is the infection, then we can do virome analysis to identify which virus is it and from the plant. So how we do it? We just do a whole metagenome of the entire plant, remove the plant part of it. For example, we are doing a virome analysis on a rice sample. We sequence the rice leaf sample if there is an infection in the leaf. We remove the rice part of it because we already have an existing database for rice. We already have an existing genome for the rice. We will remove the rice part of it from the entire metagenomic data, which will be somewhere around 80% to 90% of your entire data. Then only that 10% which is left behind will be taken for your metagenomic analysis. We will assemble, we will do the metagenomic analysis, and then we will take out the virome, which are the viruses present in that 10% data, and identify what was the infection in that particular plant. So if there is an unknown infection, if there is an infection which we are not aware of, then we can do this approach and identify what type of pathogen or what type of viruses or what type of bacteria or what type of funguses have infected the particular plant. And these are quick processes because we just extract the DNA out of it and then go ahead and do it. If you are doing it with RT-PCR, you have to design that particular primer. If it is an unknown one, you don't know what to do with it, you will sit, design, decide the primers, and then one after the other you will have to test the RT-PCR and then you have to identify. Here we can just take out the DNA and sequence it. Then improve crop yield and quality. There are many studies which are done on rhizosphere that the microorganisms help the plant in the yield, in the crop improvement, in your growth, in the plant crop development. All these stages, microorganisms play a very important role. We already know that in the roots also they play a very important role. So this study is becoming more popular with these type of applications which are coming in. Then rhizobiome is one more. Drought and salinity, stress management, biofuel production, and bioenergy crop. So all these small microorganisms are very important, and not only in the plant field, it's also important in the other fields like human, whether it is disease management, whether it is nutrigenomics, whether it is human gut. these small organisms are playing a very important role now in the application sort to identify, because they are contributing a lot. For example, in us also, if we are eating a certain type of diet, we have a certain type of patented microbiome. Now that is also coming in play, that human beings can be like, if you want to lose weight, so your microbiome will help you out if you know that what type of composition of microbiome you have. So in every field, microbiome is becoming very important. So it is very important for the plants also. The same way it works for the plants, that how they are helping them, like in the growth or productivity or so on. Specific with the viruses, there is virome. I've already spoken about it. So in virome, again, we have two sorts of viruses. One are DNA viruses and one are RNA viruses. Mostly plant viruses are RNA viruses. So we do a metatranscriptome sequencing in those cases to identify the viruses, because we want to target the RNA-based viruses. But yes, if you want to do the DNA viruses as well, we'll have to start with the DNA extraction and then go ahead with the virome analysis. So there are two sets of like virome analysis. One is DNA based and one is RNA based. Again, in this, there are also kits available to do a nucleic acid extraction, where DNA and RNA together can be extracted in a particular nucleic acid, and then we go ahead with the sequencing where we have both DNA and RNA together. But this is again, if your objective is just to target the RNA-based viruses and you want to only know that which are the RNA-based viruses, then if you do nucleic acid, you're wasting your sequence data there, right? So instead of that, we'll just go with the RNA-based viruses. We'll extract the RNA from it and then sequence it, and then identify the various different viruses which are present. So as Dr. Kumar already told, that you people have had a set of analysis which you have done, so it will be a repeat or it might be enhancing some few points. So now in 16S, there was QIIME1 which was used earlier. Now it has changed and we are now using QIIME2. There is a pipeline of QIIME2 which is being used for amplicon base, and where we are only changing the database based on which amplicon it is. If it is 16S, we use green gene or silver. If it is ITS, we use Unite database. If it is any other custom amplicons, we use different, like for example, nematode if it is, or insect, insect identification also we can do through microbiome. If that's the thing, we can also identify amplicon based on plants. If there are different sorts of plants, we want to identify which plant it is. Again, we can do an amplicon base. Anywhere where we know a target gene, we can apply this amplicon base. So in those cases, we use a custom database instead of the green gene or silver. We use a custom database. And the custom database, we make it through NCBI. And then we use that custom databases where we introduce the taxonomy as well as the sequences, and then go ahead with the identification. So how the pipeline works, initially, as I said, we are extracting the DNA. We are using a primer sequence with the Illumina adapters in it for capturing the particular region in 16S. And once we have captured that particular region, that becomes an amplicon, which we are going and doing indexing, which will separate my different samples. So we are not running one sample in one run, we are running 70, 80 samples in one particular run. So each sample has its unique barcode to differentiate between one sample to the other sample, and then we sequence it on a next generation sequencing. Once we have sequenced the entire data, we go ahead with getting a raw data, which is your FASTQ file, which we get from the sequencing machine. Now, if it is a MySeq sequencing machine, we get a 2 into 300 read length sequencing sequences, which we go ahead and then filter it out. So you can see there is a filtration step. We check that the sequences are good quality. If I mean by good quality, what it is, that the quality of each base should be good, should be more than somewhere around 25 to 30 QV. And your trimming ends, because the sequencing is 600 cycles, which is 2 into 300, your end of the sequence of R2 are usually poor bases, but not in all the sequences it will be like that, but majority of them, the mean or median of that particular end of the sequences will be dropping to somewhere around 20 or will be close by 20. So we want to trim that, only those part of the reeds, so that my amplicon which I am going to stitch and make should be a good quality amplicon for identification of the bacterial species. So what we do is that we do a fast QC and then we do trigonometric for trimming, quality filtration for fast QC and filtering out the reeds. Then we stitch the reeds. How do we stitch the reeds? There are various different tools like PandaSeek, Flash and other tools where we stitch the reeds, because we have 2 into 300 and my amplicon size was somewhere around 450 to 500 bases. Now if I have to stitch the reeds, there is an overlap of 60 to 70 bases in between, and we can stitch the reeds and make the amplicon. So now each reed has been stitched and made into amplicon. Now this amplicon, each amplicon will be used for my identification, and each reed which is getting aligned to the bacterial abundance will be calculated based on the algorithm, that what is the abundance of it. So what I am doing, that I am making one reed as stitched reed, so that I have a full amplicon sequence of it. After that we use data 2, this is QIIME 2 which has been introduced newly, where we are doing a denoising. Denoising is nothing but we are removing those tags which are redundant, which are not good, and removing those sorts of reeds which are stitched but they are not good to go for the abundance. Then we are using QIIME 2 pipeline. There are two segments here. One is for doing your taxonomic classification, and another is for your statistical analysis. And again in 16S specifically, there are new tools which are available, which I will be talking, for functional abundance as well. Now in QIIME 2 we do a representative sequence search. What do I mean by that? Anybody can tell me what is the difference between QIIME 1 and QIIME 2? You have run the pipeline, right? You're not run the pipeline. So you have heard about QIIME 1, QIIME 2? No, okay. So there was earlier a method of QIIME 1 which was used, which was based on OTU. OTU is operational taxonomic units, and now what we are using is ASV. Yeah, now ASV, anybody can explain what is the difference? What is the difference between OTUs and ASVs? Somebody has to be louder enough for me to hear. Perfect. Yes. So OTUs are the ones which are formed after clustering. What happens is that we cluster the reads and take a representative sequence out of it, right? Which we were doing in the previous QIIME 1. That's the reason that is said to be not that precise. So they have come up with a precise method, that is ASVs, where we are not using this clustering method. And ASV gives you a better abundance. That's what is the claim. So now, as it is, QIIME 1 is absolute and we cannot use it anymore. We are not supposed to use it anymore, but though we are using it wherever we don't get good abundance in QIIME 2, so we switch to QIIME 1 because we are more used to the QIIME 1 pipeline and we know in and out of it. But we are learning in the process of QIIME 2 as well. So we do ASV identifications here. Once we have the ASV with us, we have a feature table From the feature table, we can do lots of different statistical analysis like differential analysis. These are new sorts of analysis. Differential analysis will tell you between your samples, like how we do it for RNA-seq. We have differential analysis where we talk about different expressions of genes. Here we talk about different samples having different type of bacterial abundance. So how one sample is differing from the other, we can know from the statistical analysis of differential analysis. Then we have PyCrest2, there is taxa for fun. These are specific for amplicon based 16S analysis where we can do functional analysis. These are new tools which have been developed which can do the functional. So earlier it was like if we want to do functional at all, we cannot do amplicon based methods. But now we can have a superficial idea of the functional through 16S as well using PyCrest2 or taxa for fun, which gives you functional analysis, will give you KEGG pathways, will give you what is the difference between metagenomics and 16S is that in 16S we are capturing a particular region. Then how do you get your pathways? Anybody? We have that slide. We'll go to that slide. How do we get our, because we are only sequencing a very small region of a gene and a pathway. If I have to tell a pathway from there, I don't have rest of the genes to tell you. But still they have come up with a pipeline which can tell you based on simulation that how the pathways, which pathways is abundant in that particular sample. But still if your objective is based on functional, still shotgun metagenomics will be an approach to take. Here you can have an idea when you are doing 16S. Then obviously we have taxonomic classification, which is the heart of 16S, where we can identify the abundance and take out the different organisms and what are the percentage of organisms from your phyla till your species level. So all your taxonomic classifications can be done. And then we can go with heat map, corona plots. These are all plots which tells you or which helps you to interpret your data. And again we have from the biome file we can go with this pie charts where you can do sample wise pie charts and heat map when we want to compare different samples with each other. So we have already covered what is the difference between QIIME1 and QIIME2, so we will skip that. We'll go to the PyCrest. Now in this PyCrest analysis, anybody can tell what is the difference? Is it visible? Okay. Anybody can tell? Anybody has worked on PyCrest? Anybody has seen the results of PyCrest? So earlier again there was a PyCrest, now there is a PyCrest2. Because earlier we had OTUs based method, now we have ASV based methods, so they have also come up with PyCrest2. Anybody who can tell me? Then we have 16S and that's a small region of 16S, how we are able to do the identification. Is it visible? Anybody can tell me. Nobody? Okay. So there is a stimulation. So if you can see here, we use a reference tree and multiple sequence alignment. We have already received the ASV at the stage when we were doing our 16S. We already have received our biome files. Right? When we are doing 16S, we have the ASVs with us. We take those ASVs and map it to the gene family. There is a database which is created for a gene family. We map those ASVs against the gene family of a reference database which is available in these tools. Once we do that, we have a predicted gene content per ASVs. Here we are doing a stimulation. We have a predicted gene based on the database which is available. And based on that, we are identifying my KEGG pathways. That's the reason I'm saying it gives you an overview of the pathways. It will tell you that these are the pathways for metabolic, or these are the pathways for carbohydrates, these are the pathways of so and so lipid metabolism, whatever. These are the pathways which are available. But always remember there's a small region which we are sequencing, and then we are identifying that. So there is a simulation which we are doing here. We are aligning it with the gene copy per reference genome, that is the ASVs are aligned against the databases. Then we have the predicted gene content per ASVs, and we have the ASV abundance, which we did it previously also, right? The same way we have now gene-based abundance of ASV based on the reference database. And then we are predicting the gene families, which are gene family profiles. So you can see they're all talking about gene families, not genes. They are all talking about gene families, gene family profiles. These are big terms, right? So it gives you an overview idea. It doesn't give you the actual idea. If I have to get the actual idea, I have to go for shotgun, where I'm doing gene-wise. All my genes are there which are getting sequenced for that pathway, right? And then only I can tell that this pathway is there, and in this situation it is there. Here we are just doing a superficial so that we know which are the pathways which are abundant, but gives you an overview of it. Then based on that, we predict the pathway abundances. So what we are doing, that we predict the particular ASVs. From ASVs, we are aligning with the gene families which the database is present, and then we take the representative sequences from ASVs against the gene abundance. So each ASV abundance is calculated based on the gene family, and then we identify the pathways. So if we have a small region also, we are able to fetch the pathways based on this particular matrix. This is a matrix from PyCrust 2, right? And then we can get your KEGG pathways and various different. And yes, earlier we had PyCrust 1. Now we have PyCrust 2. PyCrust 2 is more efficient enough because they have made the steps based on ASVs. So the PyCrust has also evolved, and now we are in a better situation than what we were in previously. There is a similar tool of Taxa for Fun, which also does similar approach. So PyCrust 2 and Taxa for Fun are for functional abundance or functional classification in 16S only. So if you are doing a fungus sample and ITS and then you want to do functional, that's not possible because these are based on your 16S chemistries. Now coming to the whole metagenome, if you have any questions for the amplicon side, you can ask me, or else if you want to ask me at the end, I'm fine with both the ways. No questions, okay. So whole metagenomics. Now in whole metagenomics, as I said, we take the entire DNA and sequence the entire stuff. When we are sequencing the entire stuff, we have lots of things in the sequences. So there are different approaches to handle this. One is that we have, like for example, we are using a plant sample. We will have a huge content of the plant which will be there. But what we want to see is the microbes which are there. So what we will do, we will remove the plant content, that is contamination removal. In our case, when we are trying to take care of the microbes, plants becomes my contamination, but the contamination will be more, because when we are sequencing a plant sample which is a like infected plant, whether it is a bacterial infection or whether it's a viral infection, I will have the plant DNA more than your microbes. So your majority of the sampling data will be plant, which we will remove, and then rest of the data we will take for analysis. So what we do is, again, we do a filtration of reads. The only difference is that we don't stitch the reads here. Why we don't stitch? Because there is no amplicon here. We don't have an amplicon which we have to make and then we have to go for the sequencing. So we take the entire reads for de novo assembly. We assemble the entire reads. I hope everybody understands what's an assembly, right? Yeah, so we do a de novo assembly. After the de novo assembly, we predict the genes. So this de novo assembly, we expect that everything is there, whether it is virus, whether it is fungus, whether it is bacteria, everything is there in this de novo assembly, and we directly go and predict the genes. Once we are done with the prediction of genes, we go with taxonomic classification at one end, and the other end we go with functional classification. In taxonomic classification, we again use databases. There are tools like Kaiju, there are tools like other tools where we can get the taxonomy. You can also do this particular whole metagenomics with KIND database. Quine2 also does whole metagenome analysis. We can use that and use the databases which are available, or else we can do a blast against NCBI. If nothing is there, we can blast against NCBI and get the taxonomic information. So from phylum till species level, we get the taxonomic abundance of different bacteria or fungus based on what databases you are using. And at the functional end, we can use again Cognizer or other tools for doing the functional classification, wherein we will get different KEGG, COG. And again, if you are like interested in doing EMR, that is antimicrobial resistant genes, there are databases available for that also, where we can do antimicrobial gene resistance through whole metagenomics, right? So when we are doing whole metagenomics, we can get a deeper insight of the particular samples along with other components of functional categories, where I know which gene is going where and which pathways they are associated with. All these informations can become because we have the entire DNA which is there. And AMR is very important part in this antimicrobial resistant genes, where we can talk about the gene classes, gene families, drug classes, which are the AMR resistant genes in the particular samples. We can identify that, and it's just a simple blast against the CART database. There is a database for AMR, that is the CART database, which can be used for identifying the AMR genes. From the metagenomics, how we can do the virome analysis. Now after COVID, the entire shift for research has gone to the viruses. So now from metagenomics, there are approaches which are coming out to do virome analysis. Now when we are talking about virome analysis, till de novo assembly it remains the same, where we are doing a de novo assembly, we filter the reads, we do a de novo assembly, we have everything in this particular sample. Then we do a gene prediction, and once the genes are predicted, we take the genes and blast it against the respective databases of viruses. So because virome is not that a well-studied, databases are not that extensive, specifically for plant-related viruses, the databases are not that extensive. So what we do is that we take different sets of databases, like we fetch from RefSeq, we fetch from different databases, and we fetch from RBDB as well. We map on all the databases and then see what are the concordant results coming, and based on that our entire interpretation of the last virome analysis sheet, which is your viral abundance, we decide upon. So what we do is that read level, we have different steps. We can do it at the read level. We can directly take the reads after removing other contaminants, like plant contaminants or any animal contaminants we can remove, and then we take it and blast it against RBDB, or else at a read level we can do Kaiju. Kaiju is again Kaiju also has an online as well as a standalone version which can be used. So in Kaiju database also we can take the viruses as well as like we can take the reads as well as your genes to do it. So we do it at a gene level, we can do it at a scaffold level. After the assembly you get the scaffolds. At the scaffold level also you can do this particular analysis, and at the gene level also you can do this particular analysis. So this particular analysis can be done at each level to check, because the viral databases are not that Like it's in the learning phase and we do not have an extensive database specifically with the plants. We don't have an extensive databases of viruses. So we have to do it at different levels so that we know that the results are concordant at each level. And the pipelines are also not that extensive. So here we are using a blast, simple blast against different levels, like at the read level, scaffold level, gene level, and different databases to know that the results are concordant at each stages of it. And according to that, we report the virus, which are the viruses and what is the percentage of viruses which are present. Apart from that, you cannot get the taxonomic classifications because viruses are not well classified across the phyla or across the taxonomic classes. So we use virome analysis in this fashion, and we are using a homologous based approach, which is your blast, which we will be doing against each of the databases. So the virus databases look like this, where we have 0.5 million viruses which are reported in Kaiju, which includes your plant, human, and rest of the viruses. Again, RBDB is a very extensive one with 0.8 million sequences, and your NCBI is the maximum one with 1 million sequences deposited in it. So we use all these databases to classify our virome databases. Anybody has any questions till now? Is it a bouncer, or is it that I am such a good lecturer that you are able to understand everything? Huh? Okay. Yeah, if you be interactive, I also get rest. Ah. Fine. So how? So we have spoken about the pipelines, how they look. What are the pipelines? So these are all standardized pipelines, which mostly all the industries are using. Some bits and bits here and there based on your objectives. Rest the pipelines are standardized, and these are, you will get it online as well, but you will have to optimize it based on your results. So the results after what we get after doing all this analysis is your taxonomic abundance, where you can get your percentages of abundance. You can make a pie chart, you can make any other plot which is there. When we are talking about metagenomics, we have taxonomic abundance across the organisms, like we know whether it is fungus, whether it is overall taxonomic abundance of various different phyla which are there. And then we talk about the bacterial specifically in whole metagenomics, then fungus, then viruses, based on your objectives again. But when we are doing 16S, we are only targeting a bacteria, and we talk about abundance only of our bacteria and rest of the things. Again, Corona plot is one more representation where we can have an interactive phase, see different levels with an interactive graph. At functional end, we get the COG classes, PFAM, KEG for the metagenomics one, whole metagenomics. But when we are talking about 16S, as I said, we have KEG COG from the Picrus 2 or taxa for fun. We can also get the abundances from there, and we can represent in different manner like pie charts or any other plots. So where the research is going on at present with the microbiome, and what is the future for microbiome. This is an important slide where how the crop production relies on the pesticides to manage the diseases, and also the biofertilizers. Like we are using lots of chemical fertilizers now, but we are seeing in the market, you can see there are many biofertilizer companies coming up. What is the approach they are using? They are doing a basic research on these microbiomes of different areas, different plants, different crops, different diseases, and then they come up with the good bacteria, so-called good bacteria, which help the plants in being a disease resistant or in being crop yielding plant. So the yields can be increased with this biofertilizer using these microbiomes. So we do not have to rely only on the chemical fertilizers anymore. There is other rhizosphere engineering also going on, where we can introduce the microbes in the soil of that particular region and then increase the particular yield of the plants. So these are different applications where microbiome is helping in such. And if you see the last one, rhizosphere can serve as a candidate taxa for biofertilizer and growth supplements for the crop. Then microbes mediated nutrients uptakes, disease resistance, stress tolerance, and some examples of microbial functions crucial to the agricultural production systems. So how these are there, where we can understand the functionality, we can understand which are the genes which are going up and down in these bacteria, how they are interacting, and what exactly is happening with that particular crop. If we know this results, we can do microbiomes to understand all these sorts of things. Then we can exactly go and target these particular sectors where we know that stress tolerance, or we want to develop a disease resistant crop. So we can use microbiome techniques. It's not only the genotype of the plant which is involved, there are external factors also which affect, which is the research which we are at present. And so lots of microbiome, already India is having lots of government initiatives in microbiome with respect to, I believe, humans at present. But if we come up with this type of research, then obviously plants will also one of the microbiome projects which we can think of. Then antimicrobial resistance is one more area which is talked about, but not much on the plant sector. But this is a very important factor for the antimicrobial resistance, which we deal in the hospital samples, with the human samples, where we are able to identify. So through metagenomics, we can take out the antibacterial resistant genes and tell that these are the antibacterial resistances against these genes, which are the classes, gene families can be talked about, the antibacterial resistance drugs can be talked about against which the resistance is. So there are databases which we can fetch information for these type of results, and then we can give abundance. In this also we can give an abundance on each sample, what level of antibacterial resistance is there and against what it is. Then I believe I'm done with it. So thank you. And if you have any questions, you can go ahead. Yeah, go ahead. You have this short from there.
Thank you so much for this interesting lecture.
Thank you.
Ma'am, I have a question from metatranscriptomics. If I am doing metatranscriptomics shortcut instead of sequencing, can I use the data and get ASVs?
From metatranscriptomics. See, ASV, what do you understand by ASVs?
In metatranscriptomics also we do that we have that 16S gene is also getting sequenced.
But do you have the guarantee that all organisms 16S has been sequenced there? So for metatranscript, first thing is that why do we use ASVs? We are doing it for classification, the abundance. When we are doing metatranscriptome, what is my aim? I want to know the gene expressions, right? See, QIIME 2, if you are using QIIME 2 for ASVs, does it do metatranscriptomics? Will it give me a differential expression of a gene? Then why do you want to do ASVs there? you have sequenced the gene, the transcriptome of the gene for the expression of the gene. Then why do you want to do this? So in that there is an approach. There also you will get abundance. The abundance will be based on your, you can do it with NCBI database. There is Kaiju database which you can use. So what you can do, you can assemble, you can predict the genes and take the gene for expression, take the gene for abundance. Because you will not have similar genes which are sequenced in all the organisms. Your results will go wrong if you try to target a particular gene and do. No, so there we are taking out the entire DNA and going ahead, right? Because we don't want to miss out on any other organisms which are there, right? So along with the plant we go and sequence it. But later on in analysis we are first filtering out the plant part. If the genome is not available for that plant, we make a database of the plants which are available in the family level or in the order level, and then we map, we remove that particular contamination and then go ahead and just analyze the rest of the things. Right.
Why are we doing post-contamination removal before assembly? Like since we take the genome from any database, that would be assembled, right? So if we do contamination removal before assembly, will that not make any difference after that?
So your assembly step takes time, right? So if you remove your, and after assembly if you are removing stuffs, there can be a possibility that your the bacterial reads and your plant samples can be assembled together because of misassembly.
If the whole genome is not, like if we remove the contamination before assembly, is that any possibility that there will be misassembly after removal?
No, so what we are doing, we are removing the unwanted thing in the beginning only. We do a read filtration and then we map the reads to the reference genome. We are using a mapping step there, right? After assembly, if I have to remove, what you will do? You will do blast. You will do multiple sequence alignment. You will do something like that. You will do a blast. Usually we do a blast, right? In blast, will it be taking the entire stretch of sequence and blasting it? It will blast in parts. Is my objective that? No. I want to remove the entire stretch of reads. So what I will do a mapping. I will remove the contamination and go ahead.
It's good that she is asking this question. I'm sure this kind of doubts must be there in everybody's mind, at least those who want to learn. What Akshita is trying to tell, in any sequencing project, data curation is an integral part of any project. The data curation means two objectives. One is poor quality reads from the data what you got from the machine. That is judged by, Akshita already told, quality values. That's number one. Number two, the read which is not belonging to your sample. For example, if you are addressing a whole plant and microbe interaction as a target, certainly the plant would contribute to the reads. Reads, I mean the nucleotide reads or RNA, whatever. And those reads would become a kind of noise in your data. This noise need to be removed. If too much noise is there, can you sleep peacefully? No. You have to silence them. And the best way to silence here is to just remove them. So the noise remover is indeed an integral part of any genomics downstream program. And what she is telling, the plant DNA, if you are targeting a microbe as a target, and they are noise, and this noise needs to be removed. If you remove it, you end up in a data which is curated data, cleaned data. And this cleaned data is very easy to handle. And you can apply your bioinformatics I'm not singling it out. I'm sure many of you must be having the same doubt why we should remove the plant DNA part. We have to remove. And the reverse also. If you are addressing the plant as a target, you have to remove the microbial roots. And there are ways and means people have developed. There are specific signatures. Using those signatures, you can very comfortably remove all those leads. For example, mitochondrial reads when you are addressing the prokaryotic genome or the prokaryotic metagenome. Then you have to remove them. So there are multiple ways of doing it. For example, some certain regions would selectively amplify the bacterial genome. We can use those markers. But sometimes you may end up getting, some mitochondrial chloroplast genius inflammation, and we have to remove that. So remember, noise removal, or rather the non-target sequence removal, is an integral part of the sequencing effort, and this is what is called as a cleanup step, right? And the cleanup step, I think you recall, the team from Netherlands, they clearly told the importance of cleanup. The whole process is called curation, evacuation. There are several steps. You have entering the removal of the poor quality base calls, or correcting the poor quality base call based on the reverse read. Several steps are integrated. Yeah, questions for or other clarification from our team. Can stand up and tell bit louder.
Right. Yeah, just adding on his point is that when you're doing a amplicon and you have mitochondrial or you have chloroplast contamination there is a step in quine. In this case, we don't have to do it on the reed because we are stitching the reeds in the beginning, right? There is a step in quine itself to filter out these mitochondrial. You can just mention there that filter outing the mitochondrial or chloroplast. The entire mitochondrial or chloroplasts are filtered out in those cases. And then you proceed with your analysis where you will only have your microbial abundance.
In continuation with the study, now in RNA sequencing, now we are seeing we need to see the DEX, that is differential expression genes, in both prokaryotes and eukaryotes, so it comes to the interaction. So in that case, we will have reads like in a plant we have 63 million, 64 million reads, and we come to bacteria, we get only 20 million reads. So how do you take the normalization there?
See, you have to, like for the plants, either you remove the plant separately and do a differential of plant separate and your microbes separate, right? If you have a very less data in microbes, your coverage has to be high. You have to remember that. Because your plant DNA will take up the entire 80% or 90% of the data. And if, for example, I'm doing 4 GB and I have only 1 GB left behind, right, then I have very small. But if I do 6 GB and I have 2 GB left behind, I'm increasing my smaller portion by higher, right? So we have to take a higher coverage. We can do separately the plant and separately the microbes. How do I do separately? I can do a reference base for the plant. If the plant genome is available, reference base entire process I can do. And your microbe, as it is a meta transcriptome, so I will have to do a de novo approach. I do it separately. I remove the reads. So I have a microbial reads, I have this. Then you can do the normalization, right? We cannot do that together and then normalize the data and then fetch out what is there at the end. Then you will go haywire with the entire thing. You can just simplify your data.
I have individual 16 RNA sequence for the purpose of identification. Can I use that sequence for pathway analysis?
Individual in the sense Sanger sequencing.
Yes.
How will you use that for pathway analysis?
That's what
You can use it for phylogeny and all those stuff with 16S, but in pathway there are many other genes as well, right? So you cannot take one sequence, you cannot do this approach also because there is lots of reach which are there, right? So with 16S you will not be able to.
Intent to ask is based on the species name can be pretty functional.
Okay. No, but with Sanger reads, single reads, that's not a possibility there. But we can do phylogeny, we can do, there are other things which we can do with this, but not the.
Ma'am, I have done RNA-seq analysis using a CFG workbench.
Is it a metatranscriptome or RNA-seq?
RNA.
Okay.
RNA-seq.
Of a plant.
Of a plant, yes.
Okay.
So, ma'am, I get the reads in FPKM and RKKM, that is the communicate. I need to know which one should I go for to the, gene comparison.
Okay.
So,
So you're using which tool?
CLC workbench.
You're using CLC workbench for the entire thing?
Yeah, for the entire, yes.
Perfect. So see, these are different methods. You can use either methods, right? We usually use DSeq for R. These are more enhanced R versions of this. CLC I'm not personally using for RNA-seq ever. So, but FPKM, RPKM are different methods which can be used. You can use FPKM and do this, but now new method is come TPM, right? So that's more accurate. All of this will give you a differential expression, right? All these methods, but you can use either one of them. But if you want to do you have replicates as well, then you go for DSeq2. That gives you very good results. With DSeq2, you will get your differential expressions with your replicates. You can put your replicates controlled and treated, and you can go ahead with that. And you will also get your differential expression. Normalization techniques are better, and you'll get your entire thing.
In earlier lectures, we have studied for reducing the plant DNA, they have centrifuged the sample and they collected the supernectin. So, how much it will be corrected? Whether it also required separation of plant sequences?
No, see, whether you centrifuge or anything, you will still have a contamination of plant in there, right? It will reduce it. Like if you had 90%, it will be becoming 70%, right? Or it will become 50%, right? Based on the separation. But still you will have your contamination there. Theoretically, it is said that it will not be there, but practically you will have your contamination there, which you will have to remove in analysis.
Then it comes to the sequencing stage.
stage 3 is about, at specific markers, you can remove the reads itself before you get into the assembly and annotation on those. So there are multiple stages where you can systematically remove the model sequences, so that finally what you have in the hand is a qubit sequence. Even stuff different.
For example, I will give you that in mitochondria, when we are extracting mitochondria, there are wonderful kits which are available for mitochondria, right? Plant mitochondria. We extract mitochondria and we expect that it should be only mitochondrial of the plant. But still we have genomic mitochondria there, right? But the portion will be very less. We are supposed to get only 1 or 2 or 3%, but it is there. So when we are doing a de novo mitochondria in this case, we need to be careful to remove the chromosomal contamination from it.
I would have one more point here. Suppose if somebody is doing interpreting microbiome, especially the profile, using D3D, you end up getting more than 90% of the reads belong to the mitochondria, chloroplasts, which we don't want. Right? When you do the sequencing, which you already pointed out, 4 GB of data, 3.5 GB of data belong to the plant. Only a fraction, 10% of the reads belong to the top. But that also reflects the load, microbial load. So that is good enough to come out with the hypothesis validation, whatever you want to do, but this is kind of that is how this is.
From against post-contamination in case of shotgun sequencing. So, like after removing the whole genome, like for example, wheat, in case of wheat, even after removing the host sequences, after assembly, after taxonomic classification, I found that there are some other plant species, like the genome of some other plant species, which is there in that sequences. So, is that wrong, or can I use, like, should I collect those particular sequences again and do removal of that from the first?
No, so you have done the mapping of the reads on the wheat genome and then you have removed and you are getting genes of plant base?
Yeah. Some other, like wheat species, like some other wheat.
Some other wheat species you are getting.
Yeah.
Perfect. So in these cases, like for example, you have done your de novo assembly, you have done your gene prediction. After gene prediction, which database you are using? GRAS. And GRAS is giving you your, so you can go back and filter it, or else at gene level you can remove those genes. You can remove those genes based on the coverage of the genes. Like whether the entire coverage of the gene is there or not on the plant, you can remove from there and then go proceed with your microbiome.
With respect to this virome analysis, if the plant of study, like from where we have isolated the nucleic acid, is not explored one, like without the reference, how we can proceed, or what are the strategies to be adopted for virome analysis in an unexplored crop where we are not having sufficient database or gene.
Perfect, perfect. So as I have already addressed this particular thing, so what we can do, we will have the plants homologous based. This is the entire virome database is a homologous based approach. We can make a database of the family level if the family level genomes are available, right? Or order level if the genomes are available. You take up few model organisms from there, make a database of that and map the reads on it and remove the contamination and then proceed further.
Always the closest match. The only thing is the percentage.
Yes, entirely different. You will not have 100% of the reads removed in that case. Still you will have things which will come. When you are blasting it, you will also get a homologous, right? At gene level you remove. See, when there is an unknown stuff, obviously the complexity is going to be more, right? So we just want to reduce the complexity. So I will take closely related organisms, make a database out of it, remove it. So I'm not taking one in this case. I'm taking multiple so that I remove as much as possible. but I know still it will be there. So I can remove it at the gene level.
Rightly said, but again, the fundamental question here is at what taxonomical hierarchy or else. Maybe at species level you may not get any hit, but at phylum level you may get some.
Yeah.
It's always
With-
Is there is in the pipeline we can go by
So the environment, first of all, environment, why we are reducing the complexity? So you have to see which pipeline you are also using, right? Environment, you are hitting to virus databases directly.
that's after removal of the background.
Yes. Why we are removing the background? We are reducing the data which we are dealing with, right? I don't want my plant reads going and aligning and giving me the abundance of a virus, right? So I'm reducing it. If it is there also, it might not map, or it can be mapped, because there can be a similarity, because these are small stretches of sequences which are your 150, and if I assemble also, it will be somewhere around 500, 600, 1000 bases, very small sequences, right? So what I'm doing, I'm just reducing it. In virome cases, we don't have this much complexity, because I'm going to align it with the viral databases at the later stage and fetch out the information from the viral databases. But obviously, I will try to remove as much as possible at my initial stage itself, right? So that I have a good data for my abundance, because everything revolves on the percentage of abundance, and that comes from your each read which is there. If I have additional reads there, your abundance goes for a toss.
To add to that, I just want to know if there is any specific presently available technique to go for a sequencing of repeat region at a single go.
Repeat region at a single go. We can do it, no? But how big is the repeat? That is what is there. Like, what complex repeat it is. See, long sequencing technologies can handle repeat to an extent. So you can sequence it. But if it is a complex repeat, your polymerase will slip off. So we need to know in this particular thing, there is not a direct answer, but we need to know how complex is the repeat, how long is the repeat, and what type of.
We are having 30 repeats. For example, we are having 30 repeats of around each repeat is of around 150 or 200.
Long read. So long read, you can go with PacBio in this case. You can try PacBio. Nanopore also can do, but will matter on the repeats, how the repeat is. Which organism it is? Pardon me? Okay. Then you can easily do it with this also, Nanopore. Long read technology can help you in sequencing it. And this repeat, if it is 30 bases repeats, they can sequence it, because they are sequencing the entire stretch. But if you're doing a SNP or anything, then Nanopore is not the recommended one. Like if you're trying to do any variation test or anything for any other application, Nanopore has an error rate as well. So that has to be kept in mind, because it has an error rate of 7 to 8% at present.
Okay, any more questions from anybody? Again, okay.
Then what kind of contaminations we can see in the rhizosphere?
You can see many, like your rhizosphere, how close it to the plant. If the root samples are there in that particular soil sample, you will have the plant contamination. You can like if you have the root particles there where you are doing the extraction, obviously it will have the root stuff. But rhizosphere, you will do a shotgun one, or you will do an amplicon base. If you are doing amplicon base, then you can straight away do a 16S or any other amplicon. And if there is a plant contamination, you can just remove it. See, in amplicon, you are targeting a very specific region, right? If there is a contamination, you can remove it. When it is whole metagenome, it's a huge data which we are talking about. Like if it is mitochondrial chloroplast will come, but that's not a huge troublesome. There will be a percentage of it for the soil.
Any more questions?
Second, in curiosity, I would like to ask, like, what is the principle to separate out mitochondrial and chloroplast sequence from bacterial sequence? Like, they have some similarity. So, what is the
No, there are databases. As I said, like when we are doing QUIME, what it does, ASVs we are selecting, right? When we are doing ASV, it will assign as chloroplast. It will assign as mitochondria from the databases.
how they sense this
Through the databases, we already have chloroplast sequence, we already have mitochondria sequence there, right, for the plants. From that databases, it fetch how we are identifying which bacteria is what,
Specific
how we are doing.
sequences are
So,
there.
same way we will be identifying what is chloroplast and what is my mitochondria. And that will be given as taxonomy classification itself at species level, it will be written as chloroplast, it will be written as mitochondria. So once you do the entire taxonomy, you will know that whether it is chloroplast or based on the sequence database. You are using only one database, right, in Amplicon, either green gene or a silver database. In those databases, it is defined as chloroplast, it is defined as mitochondria.
I can answer him in a simple analogy. In a simple analogy, say microbiome data set, you are segregating Pseudomonas from Xanthomonas from Bacillus, right? The same like it is segregating it from mitochondria. Simple.
Yeah, how it is doing for bacteria, same way it will do for mitochondria. It's just that we are filtering out.
Any more questions? The last one? No? Huh?
Sir, just to out of curiosity, ma'am, when you go for sequencing in a single plasmid and a single like targeted amplicon, in the chromatogram, if you have certain unhided peaks, so
Unhided peaks, mixed peaks.
uniform peaks, mixed
Ah.
peaks. So just suggest us what are the ways to troubleshoot it.
You repeat
But-
the sequence.
No, ma'am, it's a fill.
The sequencing failed actually.
No, but so if it is a mixed one, yeah, there is an impurity, there is
The raw
multiple
material is
stuff.
very bad. However curation you do, it's not going to help you.
See, there are peak enhancers, but if you have a complete mixed data, nothing will be working.
That's what I'm telling you.
Obviously, there is something else which is having another peak.
Right?
Or else you have some deletion or something there against the reference if you are doing it.
So many times, it's a very good question. Many times, rather than spending time to curate it, it is easier to do the resequencing. Exactly.
Right. Yeah.
Okay, if nothing works, you change the product. Very easy.
Committed project. Yes.
Okay, I think it's an excellent presentation, there is no doubt, and very excellent interactions. I'm really happy for that. A lot of questions. I think many questions have popped up now. I think you have the comfort zone here, not in the last week, right? It looks like. No, they opened up and again went for a silent mode. I don't know why. Anyway, fine. Thank you, Arpita, for
Thank you.
taking out your time.
Thank you.
I think we have to give a big thanks to Arpita for being here and enlightening us about the industry perspective and the various. I think she covered everything. Nothing she done. So thank you so much
Thank you.
for coming here, making a wonderful presentation. We will keep troubling you. And yeah.
No, it's always a
I
pleasure
always
working.
tell that drama continues, character changes, few characters are same. So it is all like that only.
Yeah.
Thank you, and
Thank you.
action, I think Ramakrishnan sir.
I will just keep this here.
Somebody can take picture also and put it in the link.
Thank you. Like, it's always wonderful working with Dr. Kumar on various different projects.
Somebody can take a nice picture and it is, we'll put it in a link and social media platforms. Thank you so much. I think with that we conclude the forenoon session. Let's break now and go for lunch. Come back by 2:50 or it's 1:30. Yeah, I think people come very fast and fall on the road. You can come between 2:15 2:30 also. No problems. Yeah.